On genomics, and I'd like to call for John Swenson talk first.
Hi, thank you all for being here, and thank you to the tech team for all of your help. So all of us in this room, I presume, care deeply about effective conservation and sustainable fisheries management. One of the greatest scientific tools that we have to help us achieve these goals is population assessment. Population assessments pull together available biological and ecological data to provide quantitative estimates of population status and risk, which can then help inform conservation management decision-making, such as listing on CITES. 3 parameters that sit at the core of almost every robust population assessment are population abundance, mortality, and population structure, or stock structure. Unfortunately, these 3 crucial parameters are very challenging to estimate for many elasmobranch species, which hinders our ability to reliably assess their populations. But the good news is that it is recently feasible to estimate all 3 quantities in a single experimental design using population genomics coupled with a technique called close-kin mark-recapture, or CKMR. For those unfamiliar with the method, CKMR is a statistical technique whereby we use genomic data to assign relatedness among pairs of sampled individuals. And then we use the ratio of related versus unrelated pairs in a modified mark-recapture framework to estimate abundance and mortality. To build a little intuition of how it works, let's imagine that we have 2 populations of sharks, one that's large and one that's small. And let's imagine that we take 7 samples from each population. If we count the number of relatives in each one, we have a single relative in the large population, and we have 5 sampled relatives in the small population. This is obviously a contrived example, but it demonstrates the fundamental principle underlying CKMR, which is that for a given number of samples, we expect to find more relatives in a small population than in a large population. There's a lot of math behind this, but that's how CKMR works intuitively. A crucial caveat is that CKMR can only provide us estimates for the adult portion of the population, but this is often the hardest life stage to study for sharks and rays, so the method fills a key data gap. And it's also worth mentioning that the abundance estimate we get with CKMR is not an abstract quantity, it is absolute adult abundance. So if we want to apply CKMR to a population, what we need are lots and lots of samples from a single interbreeding population. But if we also have samples from other locations, then we can use the same genomic data that we use for CKMR to evaluate stock structure. We can estimate genetic stock structure using established and evolving population genomic frameworks, and demographic stock structure can be inferred from the distribution of related pairs. As part of my PhD, I applied these approaches to estimate stock structure, abundance, and mortality of white-spotted eagle rays within and surrounding the Gulf of Mexico. And we focused on eagle rays for a few reasons. First, a 2021 IUCN Red List assessment determined that the species is globally endangered and highlighted population size, distribution, and trend as major research needs. In addition, even though the species is protected in the waters surrounding Florida, it's not protected throughout the Gulf, and there's ongoing fishing pressure, particularly in the southwest region. At the same time, as a practical consideration, thanks to long-term efforts by Kim Bassels-Hull and folks at Mote Marine Laboratory and other collaborators in the region, when we envisioned this project, we already had the samples that we needed to evaluate stock structure and apply CKMR. So to breeze through our methods, we used restriction site-associated DNA sequencing to genotype all of our samples, and after filtering, we had 5,986 SNPs for our stock structure analysis and 3,271 SNPs for CKMR. Beginning with stock structure, these are our sample locations and the sample numbers that we used for our stock structure analysis. We had way more samples from Sarasota, which represents the eastern Gulf of Mexico, but we subsampled down for this analysis to keep the sample sizes approximately even. And this plot shows the results from a discriminant analysis of principal components, or DAPC. And you can view it like a PCA plot, where each point is a sample and they're colored by sample site. And this plot shows clear genetic differentiation between Belize and our other sample sites, And though there is overlap among the rest of our sites, samples mostly cluster by site, which suggests that we have at least 3 to 4 distinct genetic stocks within and surrounding the Gulf. We did not detect any close kin across sites, which would suggest limited demographic mixing, but our sample sizes, take that with a grain of salt. We might find something different if we had greater sample sizes. But given that each site appears to harbor a genetically distinct population, we focused our CKMR analysis on the eastern Gulf of Mexico, where we had 558 samples from Sarasota Bay that passed all of our filtering criteria. When we assigned relatedness among these samples, we identified 14 half-sibling pairs and 6 parent-offspring pairs, and we used these as input to our CKMR model. For those that are mathematically inclined, this is the model formula we used, and I know it looks like a lot, but this is actually a pretty basic CKMR model. And I wanted to put this up there first to show that there is mathematical backing behind CKMR, but also to highlight that we, in our model, added this parameter omega, which accounts for sex-specific differences in migration and reproduction. And what this parameter does is it means that the quantities that we're estimating with CKMR do not refer to the total adult population, but it refers specifically to the breeding adults in the eastern Gulf of Mexico. And so we ran some simulation testing, and then we fit this model to our data, and ultimately estimated between about 9,300 and 28,000 breeding adults in the eastern Gulf of Mexico, with a mortality rate of 5 to 30% per year. For reasons that are too nuanced to describe right now, this abundance estimate refers specifically to the year 2011, and we did not find enough relatives to reliably infer a population trend. However, if we impose a strict prior on our Bayesian model and force the population to grow or decline by up to 5% per year, which is a reasonable growth rate to assume for this species, we could have as few as 5,000 or as many as 42,000 breeding adult eagle rays in the eastern Gulf in 2026. This wide range of possible values reflects realistic levels of uncertainty, and it's not too dissimilar from other applications of CKMR. So, to summarize, what have we learned and where do we go from here? Well, we found that there are at least 3 to 4 distinct genetic stocks within and surrounding the Gulf of Mexico. And this matters because we have distinct genetic stocks in areas that are fished and in areas that are protected. We estimated between about 9,300 and 28,000 breeding adults in 2011, and if our assumptions of population growth or decline, capping at 5%, are on point, then that number could be as low as 5,000 or as high as 42,000 in 2026. And we found that mortality was relatively low, between 5 to 30% per year, with a median of about 15%. And so I hope that these numbers will be used in the next population assessment of eagle rays, but there are ways that we are actively and planning to build upon these results. We want to include more areas in our analysis of genetic stock structure to see how many genetic stocks there actually are throughout the Gulf. And we want to improve precision of our CKMR estimates, which we can do by genotyping more rays and finding more relatives. But the thing that I'm especially excited about is that thanks to efforts by Kim Bastos-Hull and Moat, we have acoustically tagged a number of rays in the eastern Gulf of Mexico, and we can fit a conventional mark-recapture model to the acoustic data and then merge conventional and close-kin mark-recapture together. To my knowledge, nobody has done this before, but I think it's an especially exciting direction that we can take with this robust data set. So in conclusion, we learned a lot about the population dynamics and genetic structure of eagle rays within and surrounding the Gulf. But more broadly, this project is an example of how population genomics and close-kin mark recapture combine to give us quite a powerful toolkit to improve our capacity to assess vulnerable elasmobrains. And so I want to thank all of our collaborators and co-authors and colleagues. A huge thanks to our funding sources, in particular the Save Our Seas Foundation. Thank you to the conference organizers and tech support team. And thank you to all of you for letting me monologue at you for the last 10 minutes. I don't take it for granted.
Okay, we've time for a quick question for John. Thank you, John.
That was really cool, and I'm really interested to hear how you think you'd approach merging the mark capture from the acoustic detections and the CKMR.
Yeah, so we can fit a conventional mark recapture model to the acoustic data, but I think the key thing that we'll have to consider is our scope of inference and making sure that the animals that we're estimating with conventional mark recapture are the same animals, the same life stages, etc., that we're estimating with close kin. And then we can integrate the two by using priors in our Bayesian model. So we can have a hierarchical model where one level is conventional mark recapture, and that feeds into close kin. Yeah.
All right, and now we'll have Stefan on stage.
Maybe now you can hear me? No? Is it better now? Okay, good. So hi everyone, my name is Steven. I'm based in Brussels where I'm finishing off my PhD. And today I'm going to talk to you a bit about the biodiversity of rays that we can find in Tanzania. So before going to Tanzania, let's speak a bit about rays and sharks generally. So there are about 1,260 currently recognized species. What's important to know is that this number keeps on increasing. So to give you an idea, over the past decade, we've been adding about 10 species per year to this list. So if this trend keeps on going, basically it's safe to say that we're underestimating the current biodiversity of sharks and rays. To illustrate this, we can turn to a foundational work that was done in 2012 by Gavin Naylor, in which they sequenced organisms from 600 species, and they found among that there were 70 cryptic lineages, highlighting the level to which we're underestimating cryptic diversity and the role that genetics can play in revealing it. And in the current situation, basically, given all of this, we're running the risk of losing species without knowing that they ever existed. And that's a bit of a sad thought. A consolation, I suppose, is that now the tools to find the species are becoming more and more accessible. And so let's get into it. How do we delimit species? So what is a species, basically? So very briefly, there are lots of different species concepts that exist, and every species concept is linked to a given property. And these properties are what we test to see whether we have the same species or different species. A lot of them are rooted in genetics, and basically what we do when we delimit species is we try to tick all of the boxes when it comes to species concepts, and basically this informs us of more or less where we are in the speciation process, which can then be linked to biological reality, which is actually what's interesting. When it comes to species ID, it often gets conflated with species delimitation, and that's a bit of a slippery slope. So ID, how it works typically is you want to take an organism and you want to be able to trace it back to the type organism that was used to describe a species. Ideally, this isn't always easy to do, and in genetics, we often do this by genetic barcoding, in which we'll sequence a gene, typically CO1, and we'll compare it against a big data set. But this relies on the data set having certain properties. Firstly, it has to be exhaustive, and all of the species in it have to be well delimited and up to date. That's not necessarily the case. And like I said, if you want to find the sequence that you can identify your organism with, it implies that the species that you want to identify is in the data set, which isn't always the case either. As can be seen by the family tree of sharks and rays to your right, in which we've plotted molecular data coverage. Enfin, they've plotted molecular data coverage. And so we see that there are certain taxons for which we don't have much data at all. This is in phylogenetic space, but it can also be applied to actual space, to geographic space. And there are some regions in the world for which we have very little molecular coverage. One such region is the Western Indian Ocean, and that brings me to Tanzania, more specifically to Dar es Salaam, which is a huge fish market, one of the biggest landing sites in the region, where I went and was lucky to spend a lot of time. And I collected loads of ray samples, 60 ray samples, and then I asked myself two questions. Well, how many species can I find here, and what are these species? So briefly, I took these samples back to the lab, and I did Oxford nanopore sequencing, amplicon sequencing. The reason why I use this is because on the one hand, we've developed a very cost-effective way of sequencing amplicons. And secondly, you can sequence relatively long reads with this, which is good because I sequenced a nuclear RAG1 gene, which is very variable, so the more of it you have, the better it is. And on top of that, I was able to sequence the mitochondrial ND2 and CO1 genes in one shot, which is pretty cool. Afterwards, I run it through species delimitation pipelines that all refer to a given species concept to have more or less of an idea of how many species I have. We can look at the results of that right here. To the left, we have a heat map of the delimitation methods. Basically, the blue squares are different species. You can also have a look at the colored stacked bars in the center of the screen that show the number of species that we have. We have 15 colors, so I collected 15 species. Now we have to identify them. And so what I wanted to do is I wanted to have as big of a database as possible. And so to do that, I wanted to collect or retrieve from a gene bank all of the ND2 genes, because ND2 genes are typically associated to type material, so the material used to describe a species, so vital for identification. And I also wanted to retrieve all of the CO1 genes, because CO1 is classically sequenced, it's very easy to sequence, and so we have a large geographic coverage of this gene. So you can make biogeographical claims with this. And the way that I did this, so I downloaded this for all rays for both genes, and then I come and I align them against my nanopore reads, which encompass both genes. This alignment then results in what we call a super matrix with quite a lot of missing data. But all you need to know is that we can use this to make a pretty little tree. And this tree we can then use to redelimit species, ensuring that the delimitations are all up to date. Then basically what the goal is to go have a look at all of our clusters that contain sequences from Tanzania and investigate them. Ooh, it's a bit. But this is more or less what we have. So in any given conspecific cluster containing genes from Tanzania, we can have a look on the one hand if we have any reference sequences. In this case, it's the very blurred star. And we can also have a look at the different species IDs that we can find in this cluster. And I have 3 type scenarios. The first one is when everything works very well. So we have, in this case, it's for Tenuhobsmeyeni, so the blotched stingray. We have all likewise ID'd sequences, and we have a reference sequence. So we're sure that our sequences belong to the species. We could then go prune off from the tree. We can go prune off the relative section, the relative clade from the tree, and have a look at phylogenetic relationships. And then what's interesting is we can then plot where all of our sequences come from, because we're sure they're the same species, and we can overlay this onto species distributions and see if everything lines up. In this case, it does, which is great. It's not always the case. So the second case scenario is when we have several IDs within a given clade. So that's one thing, but what's important to note is that we still have a reference sequence. So species identification becomes trivial. We can link it to the reference sequence. But then what we can also do is we can go prune off from the big tree the clades that contain the other IDs. That's on the one hand. And then where it becomes interesting is we can plot that again on a map. And so this is the map for Pastinacus ater, which occurs in Tanzania. And it was confused with Pastinacus siphon. And so now we can look at where the data come from and our current knowledge of the distribution of these 2 species. And what's interesting is that in the Red Sea, we have an occurrence, a sequence that comes from Pastinacus ater, and it's not yet considered to be part of its range. So this calls for further research, and it strongly supports that, in fact, the species does occur in the Red Sea. The same thing for cow-nose rays, where we have an occurrence of Rhinoptera javanica, in the Red Sea, which is also nice to know. Then we have other cases. Basically, you can cut through morphological plasticity, basically, and identify them properly. There's a lot to be said about leopard whip rays in the region. And then finally, the last case scenario is a scenario where you have no reference material in the group that you delimited in your cluster. And this can mean 2 things. Either it can mean that there are no sequences yet available for a given species that occurred once in this data set, or it can mean that there's no sequences and there is no reference material available for a species. And that's the case for, in this case, what we think is Tenuha lima in Tanzania. In fact, we find that when we go retrieve the sequences linked to references of Tenuha lima, which you can see in black here on the tree, they're very different species. So basically, we have a species of blue-spotted fantail ray, which is restricted to the Western Indian Ocean. And so when you compile everything, you have a nice list of 15 species that have been well delimited and normally well identified. And with this, I would like to, this is part of my PhD, which is a broader project, and through which I was very fortunate to be able to work with many of you, and you have supported me in many different ways, and I just wanted to say how grateful I am. So thank you all very, very much.
Great talk. We have time for one question, if somebody. No one? Oh, yes.
I may have missed, but how did you deal with the possible misidentification of species already in the database?
I'm sorry, so how did I miss?
I think there are some sequence entries in the database with the wrong species identification. And how did you deal with this?
Yeah, so every time I had to link my identifications to type specimens. So it's sequences that were derived from organisms that were used to describe the species. And then when it was the case, when I had several IDs, I took the one that was associated to a type reference, and the ones that weren't, they were wrong. For several different reasons, they weren't updated mainly. That's how I did it.
I see. Thank you very much.
Thank you.
Okay, next up we have Shaili Janssen.
Good morning, everybody. Can you hear me? Okay. So I want to start by acknowledging that I know this is a genomics section, but the talk covers community partnerships, artificial intelligence, drone surveys, and many, many collaborations across interdisciplinary fields. And I also want to acknowledge the many, many authors who have contributed to this work. So I would like to talk to you today about using genomics and artificial intelligence-based species identification and population monitoring to assess risk and resilience with respect to genetic adaptability as well as spatial fishing pressure across the Indo-Pacific. So we know from many, many studies that coral reefs harbor the richest biodiversity of marine species, and as a result, they also support a very high density of livelihoods and food security. This is seen at the highest levels in the Indo-Pacific. Unfortunately, across all oceans, overexploitation by illegal, unregulated, unreported fisheries is causing the collapse of ecosystems with respect to biodiversity and is weakening coastal economies and food security. As a result, there is a massive decline in the catch per unit effort, and this is predominantly impacting the Indo-Pacific, where a lot of the communities depend on these resources for food security and livelihoods. As you can see here, the impact on the economies in the Western Indian Ocean as a result of this is huge. When we look at the impacts on biodiversity from this overexploitation, the higher vertebrates are some of the most impacted. And in that, we know that chondrichthyes are some of the highest, at the highest level, impacted at the highest level. To put a number, about 66% of sharks and rays within reef ecosystems are in the higher threat levels. And you can see that from the declining red list index as well. And again, when you look at some of the species richness of the threatened species, again, they are very highly concentrated in the Indo-Pacific for both sharks and rays. So when we talk about the threat that is impacting most of this, almost 100% of these species, it is overexploitation by fisheries, and that is illegal and unregulated, which is caused by these reasons, but a huge part of that is unreported fisheries, because in the Western Indian Ocean, for example, about 60% of the fisheries are unreported because there's just not a mechanism to enable species-level catch composition, both for large fishing fleets as well as small-scale fisheries. So when we look at pathways to conservation, it looks a little bit like this. So you first have assessments at the species and population level for sharks and rays. Then you have protections and listings at the species and area level. And then you have enforcement, and this again requires species and population level monitoring. And then when you're looking at recovery of these species as a result of these regulations and enforcement, it again requires species and population level monitoring. But if you look at the capacity gaps, it is, if you think about a lot of the talks that have happened over the past few days, whether it be Rima's talk or yesterday's panel on listed but vulnerable, one of the highest capacity gaps that was highlighted across these talks was species level monitoring, as well as population level assessments. So these are the gaps that my project that I've been working on in collaboration with many, many people is looking to address. So the first objective is to determine species composition of catch using both genomic and artificial intelligence tools. And the second is to determine geographic origins of catch to enable enforcement of area-based management and regulations, regulatory frameworks. And the third step is to integrate these tools that we develop in research labs and in academia, and to integrate these into management and government frameworks. So I'll walk you through the work that I've been doing in a few slides. So the first step that we did to enable species-level catch composition and to address this huge capacity gap, especially in developing nations across the Indo-Pacific, is by developing this artificial intelligence-based tool which uses landing site data as a training data set. And using this tool, we can allow detailed ID using this identify mode or the live scan mode, which does a quick scan of landing sites where you have piles and piles of fish, sharks, and rays included. With this tool at the moment, we are able to identify about 30 species, including many carcharinids, with greater than 94% accuracy. The second part is to determine species composition and geographic origin. So for this, through my work over the last few years, I've been developing portable genetic assays, and that work is sort of distributed in many places across the Indo-Pacific, across the Indian Ocean, but I'll talk to you about a few examples. So the seminal work on this was done in Western India, in Gujarat, where we were looking at species composition of, in shark and ray fisheries. And here I developed this method called genome sequencing, which uses the Oxford Nanopore sequencer to sequence very quickly specimens that we find in the field. And as a result of that, we were able to identify trade and fisheries that were involved in the trade of CITES-listed species, such as the silky shark, and also identified many critically endangered species which were not identified at the species level in these markets. In collaboration with the Blue Resources Trust, we set up the first sequencing lab with BRT. And then taking this to the next level, as during my postdoc that I've just completed in the Block Lab at Stanford, I developed this genomic reference database of 2 reef shark species that are heavily targeted in fisheries across the Indo-Pacific. So for that, that required making a reference database using whole genomes and then using the genetic differences to identify populations and their origin of specimens in the illegal wildlife trade. So for that, we started with first building the carcharodont reference genomes, which were huge and revealed low genetic diversity. It then required sampling the entire distribution range for the gray reef and the silver-tip shark across the Indo-Pacific, and that looked like this. Using about more than 300 samples, we were able to determine population structure for the gray reef and the silver-tip. So for the gray reef was much more structured across the Indo-Pacific, and the structure looks like this, whereas the silver-tip was much more homogeneous. Using the genetic differences across these populations of the gray reef shark, we were able to train a machine learning tool to enable population assignment unique genetic fingerprints. So we used about 5 million SNPs to kind of train the data set, and out of that identified about 50 SNPs per population that allowed population assignment of specimens from the markets with greater than 95% accuracy. So with this, we're able to assign samples that look like this on boats and at landing sites, 2 populations, and we can tell where they came from. And lastly, I'm currently working on incorporating these tools that we've developed into management frameworks through funding and support from the Pew Marine Fellows Program and the Stanford Sustainability Accelerator. This work is right now focused in Tanzania and South Africa. So I would love to collaborate with any of you who are working there, including Stephen. And the impact of this is, of course, for biodiversity and ecosystems, but also it will enable us to strengthen economies and food security in the Indian Ocean region. Lastly, we are starting, as I said, in Tanzania and South Africa, but we plan to scale across the bio and across the Indian Ocean. With that, I would like to thank everybody who has been involved in this project, and I've tried to place them where they are. And I would like to thank the funders, and thank you all for listening.
Okay, thank you very much, Shaili. I'm afraid we've no time for questions at the moment. Okay.
All right, is Chiara around and ready? The floor is yours.
Hello, everyone, and thank you for being here. So today I will be talking about this guy and the gene flow between the Mediterranean Sea and the Atlantic Ocean. So why the shortfin mako shark? So apart from the fact that it's a cool shark, it's also the second most by-cost shark in the Mediterranean longline fishery, making it critically endangered in the basin. Moreover, when we want to manage a species, we would like to know also what is the population structure across this range. And for the Mediterranean shortfin mako shark, let's say that according to different markers used, they told us a different story, which means also that the species in the Mediterranean Sea has been always kind of overlooked in the assessments. So that's for this reason that we decided to take a genome-wide approach looking at single nucleotide polymorphisms. For this reason, we decided to use double digest rad sequencing approach, which is a reduced representation of the genome, and we use that on 6 regions. We decided to span across the Mediterranean Sea and the Eastern Atlantic Ocean, and after filtering with the Stax pipeline, we ended up with 66 individuals and more than 4,000 high-quality SNPs that helped us to infer population structure analysis and demographic inferences. So basically, the results were pretty much unambiguous. As you can see in this plot, which is at the APC, we can see that all the regions that we used completely overlap in genetic space, meaning there is literally no distinctiveness between the regions and also between the Mediterranean Sea and the Atlantic Ocean. When we look at the story of the demographic history of the species, we looked at the ancient demography of the species because of the resolution of our markers. And when we looked at that, we can see that the population has been pretty much stable throughout hundreds of thousands of years, meaning that the homogeneity is pretty much like genuine and ancient. So what does that mean for the species? So while we have this genetic connectivity and high connectivity between these 2 regions, meaning the population is called pamictic, there is also the ecological sites and what we know about the species recently. So basically, fishery reconstruction of the Mediterranean basin also told us that in the past 200 years, we have more than 99% decline in its population. And also, there is no evidence of tagged mako crossing the Strait of Gibraltar for now. So what does that mean? We thought that this could be ascribed to the population gray zone theory, meaning that the gene flow is still enough to homogenize allele frequencies, but the population is already demographically diverging. So for the species itself in the Mediterranean Sea, it means that while there is a pamictic, broader Atlantic Mediterranean population, it also means that it's exposed to distinct local pressure. And the genetic homogeneity doesn't mean there is also demographic homogeneity, meaning that hopefully we will need to manage it as a different demographic population in order to prevent collapse of it. So if you are curious about that, please reach me out. I want to thank you, all my supervisors and collaborators, and Flying Shark for making my attendance possible here. Thank you very much.
We have time for a few questions for Chiara. Don't be shy. Okay. Well, thank you. Thank you, Chiara.
My name is Geoffrey Walker, and I'm with the Save Our Seas Shark Research Center, and today I'll be presenting our genomics research into Sphyrna lewinii aggregations in the Galápagos Islands. So in the Galápagos Islands, Sphyrna lewinii form these very large, iconic aggregations of hundreds of sharks. And these aggregations are particularly important because this is a globally critically endangered species gathering at a very high concentration, and because many of these sharks are pregnant females. So these are highly migratory species that undertake birthing migrations to the coasts of Central and South America, and they swim in and out of marine protected areas, which exposes them to intensive fishing. So we conducted the first genomics assessment of this system using thousands of single nucleotide polymorphisms, or SNPs, to assess the genomic health of these aggregations and investigate the population genomic structure in the broader eastern tropical Pacific. In order to assess genomic health, we collected samples from 358 sharks in the Galápagos, aggregating sharks, as well as non-aggregating adults in the Northwest Atlantic and near Seychelles. And when we look at the genomic diversity of these three populations as measured by autosomal heterozygosity, we can see that the Seychelles had the highest diversity, followed by the Galápagos, and then there's a pretty big drop down to the Northwest Atlantic. Now, we also calculated these diversity estimates for other hammerhead species, including the great hammerhead, smooth hammerhead, and bonnethead. And so when we put this into context, you can see that the Seychelles and Galápagos populations of the scalloped hammerhead actually had some of the highest genomic diversity that we measured. And we also estimated effective population size, which is another metric for genomic health. Essentially, the smaller the effective population size, the faster genomic variation is lost from generation to generation naturally. And so we estimated it around 35,000 for the Galápagos, which was higher than both of our other adult populations. Now you'll notice that these confidence intervals are very wide, and that's because this parameter is very challenging to estimate precisely for large populations. However, importantly, the lower bounds of the confidence interval in the Galápagos Islands is about 12,500, and we generally don't start to worry about negative genomic impacts until we're down below about 1,000. So to investigate population genomic structure, we sampled young of year sharks from the southern Galápagos and from Costa Rica down to Ecuador. And essentially what we found here was no structure at all. So in this PCA, all of these dots are our young of year sharks, and we see them all forming one big intermixing genetically similar population. We don't see any other of these sample areas kind of grouping out or forming separate clusters, which might indicate genetically distinct subpopulations. So in conclusion, in the eastern tropical Pacific, we see a highly genetically connected population with relatively high genomic diversity and sufficiently large effective population size, all of which are good signs for the genomic health of these critically endangered sharks. However, one very important point I want to stress is that genomic health should not be confused with stock status. So these genomic changes occur over very long timescales for large populations with long generation times, and they don't reflect exploitation from fishing by humans in recent decades. So while this can tell us that these sharks are capable of adapting to diseases or environmental changes, it doesn't tell us anything about whether they are overfished or currently experiencing overfishing. So it's important that we are able to establish reliable estimates of demographic parameters so we can understand the impacts of ongoing fishing and protect this iconic aggregation. Thank you to all of our funders listed here.
We have time for a question for Geoffrey.
Hi. Thank you for your talk. I would like to know, how did you perform the sampling in Galápagos?
Yes, so our collaborator, Palayo, who just gave another talk in the other session, actually free dives with those sharks and has a dart, sample biopsy dart that he uses right there.
But he sampled the individuals in the ocean.
In the aggregations, yes.
Okay. And but it's like blood. What is it?
They're tissue samples that he collects with the dart.
Okay. Thank you.
Yes.
Okay, thanks, Geoffrey. Okay, next up is Giulia Maiello.
Good morning, everyone. My name is Giulia Maiello, and I'm going to talk about eDNA metabarcoding choices and gains in elasmobranch biomonitoring. The study area where we sample our data is along the Central Californian coast in the Chumash Heritage National Marine Sanctuary, who has recently been established along the Californian coast. We collected 48 water environmental DNA samples on the Cal Forest ecosystem during the spring and summer of 2023. We filtered the water we collected in order to concentrate the DNA, and then we amplified the DNA using two different molecular markers. One that is specifically designed to target teleost, but is known to amplify also elasmobranchs, and another one that is specific for elasmobranchs, but known to also amplify teleost. And we then sequenced the library using Illumina next-generation sequencing. So we compared the species that were detected by each of the two primers, and we realized that the vast majority of species were shared among the two primers, so detected by both. But among the species that were only detected by the elasmobranch-specific primer, we saw some very iconic shark species of Cal Forest Californian ecosystems, such as the great white shark and the leopard shark. We then compared the proportion of species and reeds that were assigned by each of the primary to elasmobranchs and teleost. And so when we look at the proportion, we realize that a significantly higher proportion of elasmobranch species were detected by the elasmobranch-specific primer. And when we look at the sequence reads, even a greater difference was found with a greater number of sequence reads assigned to elasmobranch by the elasmobranch-specific primer. We compared these results with a study that we did in a completely different context that was part of my PhD, but where we used exactly the same primer. And so this study was conducted on Mediterranean trawlers, and samples were collected using a passive sampler that is called the metabarcoding that I developed during my PhD. And we were surprised that from such a different environment, such a different ecological and geographical environment, we had very similar results, with the vast majority of species shared among the two primers. Some threatened and iconic elasmobranch species detected only by the elasmobranch-specific primer, and very similar proportion of teleost and elasmobranch detections. So to conclude, we demonstrate that elasmobranch-specific primer are very effective, as we would expect, to detect low abundance and elusive sharks and ray, but the teleost data returned by the elasmobranch primer are not so different from the teleost-specific primer. So what we conclude is that when elasmobranchs are the priority target of a study, but you also want to know more about the broad fish community of the area, the elasmobranch-specific primer are a better marker. So I want to thank all of you for your attention. If you want to know more about eDNA metabarcoding study we did, feel free to reach me out. Thank you.
Thank you, Giulia. Any questions for Giulia? One at the back.
Hi. Hello. Hello. Can you hear? Is it? Okay, better. Great talk. You mentioned in the later half the metaprobes. I don't know if I missed it, but for the first part of your actual study, did you also use metaprobes there? And can you talk more about your sampling schematic? Because I noticed you had a lot of sites, but I think you said you had 48 samples across all of those sites. So I guess my question is one, did you also use metaprobes for that study? And then two, how did you know how many samples to take at each site, or why you did?
Okay, so the thing is that the study we did in California was part of a bigger project that we're doing along the Californian coast, where we're sampling all the locations I showed on the map. And to sample in this location, we're using both water samples that are filtered with Sterex filters. And we're also using metaprobe attached to divers to sample eDNA, and also metaprobe casted with a fishing rod at each of its sampling sites. But the thing is that for this primary comparison, we just use a subset of the full amount of samples we have for the project, where we are mainly target the general vertebrate community, and we're using the Teleost primers.
Okay, thank you, Giulia. We'll have to
Thank
stop
you.
it there. Thanks.
Next, we'll have Rachel.
Hi everyone, I'm Rachel, and today I'm going to present to you some of the work that I'm doing for my PhD, where I'm using eDNA to study elasmobranchs in the northeast Atlantic, and I'm specifically focusing on a small bay in the west of Ireland. So to introduce ourselves, this is Ireland, and we're on the edge of Europe in the northeast Atlantic. And as you can see, we're surrounded by quite a few important shark-array areas. And inside in this red box is Galway Bay. And Galway Bay is a protected area for a number of species, but unfortunately there are no elasmobranchs included in that. And we're a little bit behind in terms of implementing long-term monitoring solutions and designated elasmobranch protected areas. So if we take a closer look at Galway Bay, these are 3 of the sites where I've conducted sampling to carry out our work. So the aim of my study is to develop a cost-effective and efficient method to monitor coastal elasmobranchs. So we sampled at these 3 sites for 2 weeks in 2024 and again in 2025. And at each sampling event, we collected 6 actively filtered water samples using Sterevex filters. And so we just collected surface water samples from the shore. Alongside this, we collected 9 passive replicates, so we used Giulia's metaprobes. And as you can see, we very simply deployed them from the coast. We also then used ELASO2 to detect our elasmobranch DNA. So here are some of the detection patterns that we're seeing so far from our data, and where this is quite a busy slide, there's a few things I want to highlight. First, as you can see, we detected 15 species from this fairly modest sampling effort, and this really highlights the importance of the bay because this actually accounts for 20% of the total number of elasmobranchs that we have recorded in Ireland. And including in this, we have 4 endangered and critically endangered species, and it's very nice to see the poor beagle detection because the Galway Bay ISRA is actually designated for poor beagles as well. The second thing I want to highlight is that we are seeing differences in our detection patterns between sampling types and locations between the 2 years. But I think this is most likely due to DNA dispersion and environmental factors rather than any changes in site use around the bay. However, we are beginning to build up a picture on how we might extract the most amount of information from our sampling techniques. So if we dig in a little bit and just focus, for another example, on what is the most effective sampling type, this is an accumulation curve showing species richness across our samples from both of our years combined. So things are kind of beginning to converge between just active alone and metaprobe alone, but it is by combining both samples, we are detecting the most amount of species. So as we add more samples and more locations and more weeks to our data set, we will begin to tease out how to get the best bang for our buck from our sampling plans. So to summarize, eDNA is effective at detecting our elasmobranchs in the bay, and we will be adding additional sites and locations to continue building this data set and investigate where the DNA might be originating from and what factors are affecting our detections. It also comes with challenges. What does this mean for our future monitoring? And can we convince anyone that eDNA biodiversity monitoring is important and effective? And even more broadly, how do we convince policymakers and decision makers that this data can be useful for the decisions that they make in the future? So please come talk to me. I've got lots more going on in the background, and yeah, delighted to talk about shark eDNA. Thank you very much.
Thank you, Rachel. We have time for one question.
Thank you. Nice talk. You had some really interesting detections of benthic animals. So I'm just curious, what was the average site depth for your sites, and were you sampling at the surface or throughout the water column?
No, just from the surface. So our water samples for the Stereovex filters, I just waded into the water and took a water sample from the beach.
And the the site depth?
Well, the total bay, like, I mean, from there it's shallow coastal areas, so the total bay I think goes to about 200 meters. And then as you go out, you reach like greater depths outside the bay.
Nice, thank you.
Okay, thank you, Rachel. Okay, and now Raksha Doddabele, who will close out the session. Thank you.
Hi, my name is Raksha Doddabele, and I'm excited to talk to you guys today about using environmental DNA to capture the genetic variation of white sharks, and hopefully use that to estimate the abundance of white sharks at local sites. I'm a PhD candidate at Stanford in the Block Lab. I began this project because there's currently no method to accurately estimate the local abundance of white sharks at specific sites at specific times. Our lab generally uses these two monitoring methods on the left to census the white sharks. So on a field day, we can count the number of white sharks we visually identify by their unique dorsal fins, or see how many previously tagged white sharks we detect that swim within range of an acoustic receiver. As you can imagine, neither of these methods fully capture the actual number of white sharks on site. So we're going to try to use environmental DNA to get there. Here's a map of one of our study sites, Año Nuevo in Central California, where all the yellow dots correspond to times that we've sampled environmental DNA at the same time as a tissue biopsy of a white shark, so we can eventually compare the DNA in both samples. As you can see in this plot, the amount of white shark eDNA alone is not really quantitative, and it isn't well explained by the number of white sharks detected by any other method. So instead, to estimate the local abundance of white sharks, we're going to leverage the actual amount of genetic variation detected in a sample. To do this, we treat an eDNA sample as a mixture of DNA from potentially multiple different sharks and see how many different alleles we detect. The basic idea is that the number of contributors to an eDNA mixture has to be greater than the actual number of alleles detected at a locus, depending on the ploidy. This method can actually greatly underestimate the number of actual sharks because multiple sharks can have the same allele. So we're going to get a little bit more sophisticated and use maximum likelihood estimation, which uses the frequencies of any detected alleles as their probabilities of detection, multiplies these probabilities together while accounting for all the possible combinations that the alleles could be detected in. You can learn more about the likelihood function used from the work of Weir and Seppi. This has already been successfully done using eDNA for round gobies, whale sharks, and kelp gametophytes, but not yet for white sharks. So to get here, I first had to find highly variable regions of the white shark genome and develop primers so I could amplify them from eDNA. To do this, I had to identify highly diverse SNPs within a narrow region of the genome, so this was a long enough region that could be actually successfully captured from eDNA, and then design and validate primers that were specific to white sharks for all of these regions. This got me a preliminary set of 5 primers on different chromosomes, which, as you can see, are highly diverse with a high number of microhaplotypes at each locus and well-spread allele frequencies. And these allele frequencies are what actually get fed into the maximum likelihood estimation model. To test the efficacy of this primer set, I simulated eDNA mixtures by randomly drawing alleles at each locus for a true number of contributors, including any masked alleles, and then compared the model's estimated number of N to the true number of white sharks. So the maximum likelihood estimation on the very left performed the best with a near exact accuracy of close to 60%. You can see that at higher numbers of true N or number of sharks, the model deviates more from what should be the correct answer, but it still provides a powerful uncertainty range for the number of white sharks in a given site and does better than random guessing. My learning so far is that even though these 5 microhaplotype loci are theoretically enough to distinguish individual sharks by their genotypes, we only are at an accuracy of about 60% for estimating the number of white sharks, so I'll have to develop more primers for microhaplotype loci to get a more accurate contributor estimation. And of course, it's hard to detect nuclear eDNA from rare species like white sharks. And my next steps are going to be to sequence these paired eDNA and tissue samples at each of the microhaplotype loci and see if the genetic variation in both samples is the same, and then apply these contributor estimation techniques on real eDNA samples from the field and compare those eDNA contributor estimations to other methods, the acoustic and visual detection. So with that, thank you so much to all my colleagues, mentors, and funders.
Hey, any questions for Raksha? No, I don't see any. Okay, thank you, Raksha.
Thank you.
Can I use the mic? I'm using the mic. The mic. Good morning, everyone. I cannot start this presentation without acknowledging the immense contribution of some people in my life. I would like to thank these amazing guys who have supported me throughout my career and my work in Ghana. They really inspired me and motivated me to go beyond my boundaries, and I would like to thank them. And to the Save Our Sea Foundation and the Marine Conservation Action Fund. Without them, the amazing work that you are doing in Ghana and even across West Africa wouldn't have been fruitful. And through their support, I am standing in front of you this morning as a Whitley Awardee 2026. And I owe, thank you. Talking about the Whitley Award, actually the women were grabbing all the award. So I, just by a stroke of magic, I also grabbed one for the male gender. Okay, so I'm Mr. Seidu, and I'm in the Department of Wildlife and Range Management of the Kwame Nkrumah University of Science and Technology, Ghana. I'm also the co-founder and team leader of Aqualife Conservancy. And this is a Ghana-based NGO with primary focus on marine biodiversity conservation. But Aqualife Conservancy, we have actually led a number of research and conservation programs on sharks, rays, guitarfish, and other marine megafauna. But today, my presentation is focusing on some of the key findings we've gotten so far on guitarfish. So my work is being conducted along the coastline of Ghana and is demarcated into 3 key regions: the West Coast, the Central Coast, and the East Coast. So the Greater Accra region, together with the Volta region, make up the East Coast. And the coastline of Ghana is approximately 550 kilometers, but we started our work along the West Coast and then gradually moved from the Central, and then we are now covering the entire coastline of Ghana. So it is a host to 3 major fisheries activities: the industrial, semi-industrial, and artisanal fisheries. But artisanal fisheries alone contribute to 80% of our total annual fish stock, and it's being operated by over 150,000 fishers along 350 landing beaches. So men are typically into fishing, while women are also typically into post-harvesting activities such as fish retailing and fish processing. And the fishermen use enormous gears to target marine species, including the sharks, the rays, the bellfish, amongst others. So we started our work in 2018, and we're focusing on all elasmobranch species. But along the way, in 2022, we published a paper. And this paper actually inspired us to concentrate more of our attention on guitarfish. And the reason why we concentrate more of our attention on guitarfish stems from three major reasons. The extinction risk of the species. According to my paper, swordfish is functionally extinct in Ghana. And the last time a fisherman actually caught the species was somewhere in the 1980s. And the wagefish is also silently going extinct. Since 2018 till now, we have never spotted a single specimen of the wagefish. And the last time a fisherman caught the wagefish was somewhere in the 2010s. So the serial extinction of the swordfish to severe decline of the wagefish means that the guitarfish is next to suffer local extinction because of their white fins. And because of their white fins, there's high reliance on the species and other sharks and rays. Fishermen are intentionally targeting guitarfish for their white fins and for their meat, which is quite lucrative in our local markets. In the baseline information, we started our work by focusing specifically on the resident fishes. And we have collected key baseline information from these resident fishes. But along the way, we realized that many of these resident fishes are now moving to become migrant fishes, moving from community to communities and then countries to countries across the West Africa sub-region. So in 2023, we decided to focus our attention on the migrant fishes, targeting guitarfish, sharks and rays. But for the want of time, our presentation will focus specifically on some of the key findings we've gotten so far on the guitarfish. So our approach, we use the snowball sampling scheme to track down a total of 58 fishes who are targeting guitarfish, and we conducted an interview to collect information on their socio-demographic, their livelihoods, their fishing activities, amongst others. We also conducted landing site survey along the coastline of Ghana, and I cannot delve deeper into them, but we recorded 4 species of guitarfish. And these are the spinback guitarfish, black-chin guitarfish, white-spotted guitarfish, and the common guitarfish that are currently extant in the country. So we gave a photo guide of all the guitarfish species that are extant in Ghana and across West Africa, and we asked the fishermen to identify. And many of them were able to identify only 4 species of guitarfish. And many actually group guitarfish into 2 categories as large and small. For example, species such as the black-chin guitarfish and the common guitarfish were grouped under one category as the large guitarfish, while species such as the spinback guitarfish and the white-spotted guitarfish also grouped under one category as the small guitarfish. And many of them, when we actually asked them to compare the catch composition of guitarfish to 10 years ago, when many of them started fishing, many of them indicated that the catch composition of the large guitarfish have reduced by a range of 80 to 90% per their recollection. And some also believe that the catch composition of the smaller guitarfish are also reduced by a range of 40 to 60%. In comparison with the sizes, many believe that the sizes have reduced tremendously and that they are now catching smaller individuals now as compared to the 10 years. And the migration pattern. In Ghana, there's a high rate of consumption of shark meats, and the resident fishes are unable to meet the market demand. So as I indicated earlier, many of them have turned into migrant fishes, moving from communities to communities and countries to countries across the sub-region. So here we were trying to map the migration route of these migrant fishes, and based on their origin and the destination as well as the frequency of catch, we're able to map 5 different migration routes. These are the West Coast migration, the Central Coast migration, and the East Coast migration. For here, the fishes actually move across the various regions across Ghana. And then the intra-regional migration. The intra-regional migration is where fishes actually move from one community after exploiting their customary or traditional fishing zone. They move to other fishing zones within the same region. And then we also have the international seasonal migration, where fishes move from Ghana to neighboring West Africa countries such as Togo, Ivory Coast, Nigeria, amongst others, to target guitarfish. When they get them, they process them into dry meat, and then they export them back to the country to supplement our protein needs. Drivers of migration. Here we find out that there are 3 key factors that pushes fishers to migrate to neighboring West Africa countries and to other communities. And these are the depletion of target species, high market demand, and low fishing pressure. Now, motivation for targeting guitarfish. When you ask them what actually inspired them to target guitarfish, you find out that the sale of the meat, as well as the fins, were the primary reason why fishers were targeting guitarfish. So guitarfish, once it's being caught, the meat is actually, or the body is actually processed into dry meat and then consumed locally. But when fishers are outside the country, then they process them to dry meat and then they export them to the country. The fins are sold to foreign nationals coming from countries such as Guinea, Senegal, and Gambia when the fishers are in Ghana. But when they're outside Ghana, they sell the fins to a middleman, which are mostly Ghanaians, who serve as intermediaries between the fishers and the foreign nationals in those countries. Now, the use of migrant fishers. As part of our interview, we asked fishers whether they have other livelihood options apart from fishing. And many of them indicated that fishing is their only livelihood stream. So we asked them if there should be a regional ban on exploitation of sharks, rays, and guitarfish, what is going to be their preferred fallback livelihood option? And many of them chose driving, which is quite not feasible. But some also chose snail rearing. And so, as part of our work, we are trying to instigate snail rearing opportunities to these fishermen. So far, we have actually recruited 45 fishers, trained them in snail farming, and given them their startup resources to start their snail business. So as part of this, we asked the fishermen to sign a contract that commit them to spend 30 to 40% of their time on this livelihood stream to make sure it's successful. We are also mapping key marine areas that are critical to the survival of guitarfish. So we train the fishermen in GPS and GPS tracking device, and then we give them the GPS together with us. We go to the various areas where they target the guitarfish, as well as the spawning site and aggregation site of the species, and then we map this area. Our plan is to establish a locally managed marine area that protect the guitarfish and the fishermen themselves, taking charge of the management of the area. And then our next solution is instigating conservation profile. We are trying to raise the conservation profile of guitarfish. And so far, we have done our conservation and awareness creation campaign in 10 communities. In 10 communities, we train fishers on safe release protocols of guitarfish, as well as the best fishing practices. So the fishermen, most of the fishermen are actually using oriented fishing gears in their quest to get abundant catch. So as part of our work, we link up with the Fisheries Commission to train them on the best fishing practices, not to use some oriented fishing gears such as light fishing, dynamiting, blast fishing. We also train some fishermen in safe release protocols of guitarfish, and then we incentivize them, especially those fishermen catching the species as bycatch. We incentivize them with a bar of soap to release the guitarfish species. Thank you for your attention.
Okay, thank you, Issa. Our next speaker will be Gobi Raj from Sri Lanka.
Yeah, good morning, everyone. So I know most of you recognize me because all the time in the lobby area. So, but anyhow, I introduce myself. I'm Gobi Raj. You can call me Gobi. So I'm from Blue Resource Trust, Sri Lanka. And today I'm going to present about from landing site to landscape, identify the potential critical habitat of the rhino pestiforms to strengthen the ISRA conservation for Sri Lanka. So when it comes to rhino pestiforms, we all know that they are the most threatened fish group in the global. And also same as other sharks and rays, they are also very extremely vulnerable for the fishing pressure. And when it comes to Sri Lanka, there are a few other sharks and rays targeting fisheries, but when it comes to for the rhino prustiforms, all are bycatch from the various type of fishing method. So yeah, for rhino prustiform for Sri Lanka, we have 8 different species, and you can see when it comes to showfish, historically we had 3 species, and the recent studies we have done. Unfortunately, last 8 years, we never recorded any of the showfish during our surveys. So we done the questionnaire surveys with support of Elasmo project in 2018 and 19. And through that, we come across this photo that in the left-hand side that 2018, one of the fishermen shared with our team member. So that is the evidence that one recent finding for Sri Lanka as a showfish. And when it comes to wedgefish, we have 2 species. One is bottlenose wedgefish, which is in the picture above the name, and the other one, bowmouth guitarfish. And for guitarfish, we have 5 different species. So you can see that picture is sharp-nosed guitarfish, and there are other 4 guitarfish, which is for Sri Lanka. So what is the challenge we're having for rhino prustiform in Sri Lanka? Unfortunately, our landing sites are more widespread, and all over the island, we have over 900 landing sites. And you can see the map where we surveyed landing spots. So we almost surveyed over 100. So it's normally around 10% of our actual landing sites. So that is a big challenge we have, because we don't have a chance to track all the landing sites, because these rhino prustiforms are more abundant in the coastal ecosystem. And also when it comes to Sri Lanka, our coastal fishery not much regulated. And so because of that, we don't have proper tracking system for the rhino prustiform or any coastal elasmo branches in Sri Lanka. And also we have good demand for local consumption of this species. So still those bycatches end up in the market for local consumption. So the question is, how we gonna conserve them? So when we decided, so it's Sri Lanka is island, and we don't have proper tracking where they are present. So we decided use GPS data, GPS devices to track the fishing grounds, which can make easier to locate their population and the habitat, and use the biological data that we collected, and then make the hotspot or the critical habitat for the species. So this project was started in 2017. We started our landing site data collection, and from that we found there are 2 guitarfishes which is more common for Sri Lanka, which is we plan to track through the GPS track. So for the field survey, we normally collect ID photograph for all the elasmo branches, including rhino prustiforms. And for the morphometric data, sex and maturity, and also length weight data. And also we collect tissue samples for our future molecular work. And when it comes to GPS track data, we hand over who are the fishermen willing to take the GPS to record their fishing location. So whoever carry, they keep this GPS on to track, record the full their fishing trip, and they hand over us returning, and then we are copying the data. And also we collect their catch data when they got elasmo branches to link with the GPS track to find where they actually located. So as a findings, so as I said earlier, like we have done a project in 2018 and 19, and then we come across our population was collapsed during the 19th century. 1990s. So after that, there's last 3 decades, there are not much, no any showfish catches. And in the right side, you can see that's the only evidence that remaining in the country that we had the showfish population for Sri Lanka. And when it comes to wedgefish, these are the two species I said we have in Sri Lanka, and both are critically endangered. And it's all around the country we are recording, but we don't have much frequent sighting in the market. And these are the 5 species from that guitarfish, and from that 4 species are critically endangered, and the spotted guitarfish is a near threatened species. So we choose the more abundant species such as sharp-nosed guitarfish and the striped-nosed guitarfish. So the first one, sharp-nosed guitarfish, a GPS tracking survey was started in 2021 in the north of Sri Lanka. And most of the sharp-nosed guitarfish, the entanglement was recorded in the crab gillnet fishery, which is 90% of the catch, and the 10% also coming from some other different fisheries such as longline, drift gillnet, and ray target fishery net. And for the maturity wise, we encountered all the juveniles guitarfishes, the sharp-nosed guitarfishes are from the crab gillnet fishery, and that's the most destructive fishing method in that area for this species. So in the right side, you can see that purple polygon, which is mentioning the ISRA for Sri Lanka. It's the joint ISRA for India and Sri Lanka. It's called Pulk Bay, and it's developed in 2024 and 25. So luckily we started our tracking surveys a bit early, and then we managed to give some data to make the ISRA to support this species. And the second species, striped-nosed guitarfish, which is present in the east coast of Sri Lanka, more abundant, and also we have in the northwest, but we started our tracking surveys in the east coast, and we recorded, and the polygon showing the ISRA where that in the east coast is called Pasikuda Kalkuda ISRA, and the intensity about the fishing intensity based on the fishing number of fishing trips. And this location, most of the fishers, they do bottom set gillnet, which is targeting mostly rocky fish, and sometimes they target lobsters as well. So these are the, this is the gear mode entanglement of guitarfish in that region. And most of the time, just juveniles, all the juveniles coming from this bottom set gillnet. So as summary, we have like the good rhino prustiform diversity for the country, but unfortunately, because of the coastal fishery, we don't have much regulation in the country, so the monitoring is very important. Unfortunately, that many landings and everything, we have to shortlist the area to manage. So this is ISRA, one of the good thing, like before getting to the law or something, we have some recorded data and the finalized output from the findings, so we can directly give it to the management team where they can actually easily go for the specific areas. So yeah, that's all. And here the India and Sri Lanka join ISRA. So this one also one of the good thing when we have nearby countries, and the regional management is very, very important. And finally, I want to thank all the funders to support our last 8-year project. And the final message, like whatever we did science, the community engagement is more important to succeed in the conservation. So we also target focusing on the community interaction to make the awareness with the fishermen to serve these valuable resources. Thank you.
Thank you, Gobiraj. We're not going to have time for questions, so you're off the hook. Our next speaker will be Brian Miranda, who's going to be talking about wide-nosed guitarfish.
Hello. Yeah. Hi, everyone. So my name is Brian. I'm going to talk about some of the work we do on the wide-nosed guitarfish in India. Just to start off, I wanted to give a bit of introduction of the species in the context of India. Basically, across several parts of India, these guys occupy extremely shallow coastal waters. And because they are occupying such shallow coastal waters, they are vulnerable to, of course, nearshore fisheries, any sort of anthropogenic activities happening. Now, within India, this species is extremely understudied, and most of our data comes from fish landing sites, and a lot of the times that's not very species specific. Coming to our study site also, this species is a low-value bycatch. Most fishers, they rarely consume the larger individuals, and the smaller ones they almost always, release back into the wild. So the study site, our study site for this was the state of Goa, which is a small state along the central west coast of India. Now, even though it's a small state, it has a lot of, variety of fisheries ranging from, persines, gillnets, trawlers, and also shore scenes, the one you can see on the top right. It's also a very major tourist destination. So it attracts a lot of tourist footfall, and because of that, there's also a lot of coastal development happening. Now, through local ecological knowledge and our anecdotal surveys, what we realized is these species are aggregating, juveniles of the wide-nosed guitarfish are aggregating in the southern part of Goa. So our goal was sort of to understand, get some more robust systematic data on what is happening with these aggregations. So we had two main objectives. The first one is estimating their population. For that, we chose 10 beaches. Now, most of these beaches are pocket beaches, so they're sort of geographically isolated with hilly outcrops. And our second objective was to understand the seasonal variation, which was mainly across 3 beaches. Now, why did we choose these 3 beaches is they show like very varying levels of disturbances from tourism as well as varying levels of fishing effort, as you can see. And Galjibagh Beach, which is the top one listed there, is also a turtle nesting site, so it already has some level of protection because of that. And it's also recently been classified as an important shark and ray area due to the presence of these wide-nosed guitarfish as well as blacktip sharks. So coming to our methods, so for both of our objectives, we basically use these walking transects methods where we walked in extremely shallow waters, about ankle level deep water, and we counted the number of guitarfish that we see, and we broadly visually classified them into these 4 size classes. These size classes were visual estimations, which we sort of validated through occasional measurements. And yeah, we also collected GPS coordinates for each time we sighted guitarfish. Just a quick video to show you what it looks like when we see these guitarfish while doing one of these walking transect surveys. They are like right there where everything along the beach is happening, where people are swimming. So that's why it's even more important to sort of understand, why they're here, and why are they choosing these sites. So so for our population estimation, we basically use this method called N-mixture models. Now, the main assumption of this method is that the population should be closed. Now, because our beaches were sort of geographically isolated, we also, in order to meet this assumption of a closed population, we did our surveys at each site on consecutive days. So it's unlikely that there is any sort of movement outside the population within a short span of time. And of course, the variables we use are rock cover, beach length, and visibility, for which we developed our own indices. For our seasonal variation was a bit more long-term. This was at the 3 sites I mentioned. We basically covered each site at least once a week over a span of 7 months, and these are some of the variables we used in our analysis. So we were able to, estimate the absolute abundance of juvenile wide-nosed guitarfish across, 25 kilometers of the coastline, and we estimated the numbers as 538 individuals, and only juveniles were used for this part of the analysis. We did see occasionally adults, but we didn't include that. So as you can see, Galjibagh Beach on the leftmost one shows, this is also the one that was an ISRA. It shows really high numbers of guitarfish, almost 200 individuals, just from a 1.5 kilometer stretch of beach. Now, there are 3 beaches here at the right that you can see. So we didn't see any guitarfish here because these beaches, we don't know for sure, but these beaches are quite rocky. So we think that maybe, guitarfish tend to prefer sandy habitats, which is why they may not be using this site. Now, the last one, Polem Beach, doesn't have a lot of rock cover, doesn't have any rocks at all, but the beach slope is very steep. So, either we didn't see them there because they might be in slightly deeper waters, or they don't use it because there's no, gentle slope for them to sort of come on the beach. Yeah, a couple of things to consider sort of in this modeling or in this methodology is that, at least for our study, the sample size was a bit lower and none of the covariates were significant. So what might be useful to maybe, use other covariates that might be affecting the distribution of these guitarfish, such as prey availability, sediment size, or, anthropogenic disturbance. And overall, it's still a very good method, a very useful method rather for, to study such behavior. Yeah, because it requires very less equipment and it's basically just walking on the beach with a torch and counting. So yeah, wherever it's extremely useful. So coming to our seasonal variation, so our 3 sites that we saw, so Galjibag Beach had the least amount of disturbance and it showed the highest number of guitarfish. And Palolem Beach had the highest disturbance and showed the lowest level number of guitarfish. So this could be that the disturbance is causing this variation, but there's also a lot of other confounding factors. So Galjibag and Rajbagh Beach showed higher numbers compared to Palolem, and both of them have river mouths near the beach. We also wanted to look at dial patterns. So as in we did surveys before the sunrise, during the day, and after sunset, and we found that they are significantly higher during the dark compared to daytime. Yeah, so we did not, and some of our other variables like tides and moon, lunar cycle, we did not find any influence. Now, tides, it might be the case that, these are pocket beaches, so the tidal influence might not be that apparent. And what we think is happening is something called as nectobenthic dial vertical migration. Basically, they are in shallower waters at night and move to deeper waters along the seafloor during the day. We did try some BRUV surveys, to sort of understand or test this theory, but we didn't, the visibility was too bad for us to even spot a single guitarfish. Just to quickly go over, some of the demographics that we noticed. So in Galjibag Beach, what we realized is in October, November, there was high number of juveniles. And as you can see from December onwards, there was a sharp increase in neonates. So either this was a pupping event or some sort of a pupping season. But we need, more consistent data to sort of confirm this. Now, Galjibag Beach showed significantly, higher number of guitarfish, and it also showed higher number of neonates. So this, it's likely that this beach is a nursery ground, and we are working on, collecting more long-term data from this site to see if they are consistently using this beach to give birth. Going forward, this method is a good way to form like a fisheries independent baseline data for vulnerable life stages that are coming on the beach where they are extremely threatened. We also want to start looking at macrobenthic diversity, disturbance to sort of try and understand why are they using these beaches and why do they prefer certain areas over the others. And of course also do some tagging studies to understand movement patterns or fine scale beach level connectivity. And we are also monitoring beaches across North Goa as well to sort of, find out other sites that they may be coming to. And we are also working with the Goa Forest Department, which is the local wildlife governing body, to sort of get them on board to also start, monitoring these endangered species. Yeah, I'd like to thank everybody, my co-authors, of course, and all the people in this photo, and all the people named there as well. And yeah, thanks a lot to our funder, Sevasi's Foundation, for funding our project and for funding, allowing me to come here and present. Yeah, thank you. If anybody's interested or if anybody has suggestions also to how to improve the project, I'd be happy to discuss it now or later. Thank you.
Thank you so much, Brian. Next, we have Eloise Richardson talking about her work with the Halawi guitarfish.
Hi, everyone. So I'm excited to talk to you about our guitarfish research jumping over to the Red Sea today. So we're all here to understand more about rhino rays, one of the most threatened groups of elasmobranchs. And unfortunately, for many of these species, there's a lack of data in regions that coincide with unmanaged fisheries or historically unmanaged fisheries. So that's where our research in the Red Sea is hopefully trying to fill in some of those knowledge gaps. The focal species for my PhD is the Halabai guitarfish. It's regionally endemic to the northwestern Indian Ocean, and it's critically endangered. And most of what we know about its movement ecology and life history is limited to the information from its better studied congeners. So there's still a lot we have to learn about this species. The limited information we do have shows that it's been observed in shallow water lagoon and seagrass habitats in the Red Sea. And it's thought that females are possibly seeking out these areas to give birth. If we jump over to the Arabian Gulf on the other side of the peninsula, they're seen in slightly more diverse habitats, and year-round use of these habitats is reported. So we have a few objectives to try and learn more about the movement ecology of these animals. One is to investigate the movement ecology across all life stages to give us a bit more of a holistic view of the entire animal's life. And we're going to be doing this using acoustic telemetry and satellite telemetry, and also considering the drivers in terms of environmental factors. So first, I'd like to introduce you to our study site. This is the Al-Waj Lagoon. It's in the northern Red Sea. It kind of acts like a mini ocean, so it experiences more extreme temperatures than the rest of the Red Sea, and at certain times of the year, higher chlorophyll concentrations. But most importantly, it's part of Saudi Arabia's Vision 2030 initiative, which means that it's a candidate for coastal development in order to bring in more tourists and attract people to visit the country. So in order to understand how this coastal development and these associated risks might affect our animals, we targeted 5 main areas in the lagoon. We captured adults using benthic longlines, and the immature individuals were caught using seine net, as demonstrated here. And I'm very happy to talk more about these methods at the end. So there's a set of standard data points we would take from every individual, and after that we would begin the tagging. So every animal gets an identification tag, and if it's a candidate for our movement study, it will also get an acoustic tag. And depending on the size of animal, we would select a different size of acoustic tag, and this would give us up to 10 years of data in some cases, monitored by an array of up to 35 receivers in the lagoon. And then finally, our large individuals would also get satellite tags. So these would be fitted with a micropat, which, all being well, will provide us up to 5 months of data on their vertical movements in addition to the horizontal movements. So after all that, for a critically endangered animal that really has very few ecological papers about it, we caught over 400 individuals over the last few years. So we were very excited about that. We categorized all the animals into 3 life stages, so young of the year, juvenile, and adults. For the early life stages, we caught almost 50-50 males and females, so that was really nice for our stats. And for the adults, we encountered quite a few more males. And so looking at our acoustic detections, first of all, this is the start of our movement ecology data, we actually managed to tag over 80 of these individuals, and we heard from 66 across the duration of the study. And as far as acoustic telemetry study goes, and again, critically endangered species, we're really excited about these results. So this is an abacus plot. Each row represents an individual, and each point on that row represents a detection within the array. So this already got us thinking that some of these animals are clearly very residential, and could this be a nursery area for the early life individuals? So this is some plots representing our young of the year and our juveniles, and we did manage to showcase that they are resident for extended periods across multiple years. So we actually have a nursery paper in the works right now. And then diving into the drivers behind this a little bit more, we can see that for these early life stages, the larger the individual, the more resident they are. There's also a partial effect of temperature, and that as temperatures are rising until about 24 degrees, so does the residency. And they're highly resident throughout most of the year, with just a slight dip in the late summer months, early autumn, which also coincides with warmer temperatures. There you are, no effect of sex. And then when we look at the adults, the story is not quite as straightforward with the residency. We do see similar patterns in that they are least resident in those same warmer months, but they actually are spending, most of the individuals are spending less than 50% of their time in the array. So being a potentially migratory species, larger individuals looking for pupping or mating opportunities, ecologically this made sense to us. And then we moved on to the satellite tagged individuals. So we tagged 14, and we managed to get data from 7 of them. So again, as far as satellite telemetry goes, we were really happy with this. And these candidates were split between winter and summer collection periods. So we have 4 individuals that were looked at in the winter. 3 males, 1 female. Each of these plots is representing the mean depth daily across their overlapping periods. And you can see that for most of them, they're spending the majority of their time in less than 5 meters of depth, occasionally taking deeper dives down to 10 meters, with the exception of our female on the top left, who takes a whopping deep dive down to 30 meters. And then when we compare this to the summer months, this was very exciting for us because although our 2 females on the right-hand side are showing similar behavior, they're detected in mostly less than 3 meters. Our female on the left did a big dive down to 158, and I apologize for the digit that's wrong there. And this is more than 3 times the maximum depth in the lagoon. When we also compare this to her acoustic data, we can see that at that point, just before June, she has left the array. So we can pretty confidently say that she's swimming around the rest of the Red Sea and up to something else. And so I'd just like to give a shout out to keep your eyes peeled for this paper coming. We're in the first set of revisions right now. This is our nursery paper, so this is focusing on our early life stages. And then just a quick plug for some of our other collaborative work that's come out. We've reported some really fun predation events, and some colleagues from Red Sea Global have also established that this area is a very important site for reproductivity, which supports our work as well. But finally, just to say, there's still a lot of work that needs to continue to give us a holistic view of what's going on with the guitarfish. I'm still investigating the adult tagging data. We also have a lot of physiology and pollution-based projects from some of our colleagues. And if you stick around another 2 minutes, you can listen to Kaitlyn talk about some of this, so please don't leave the hall. And then finally, just to say a big thank you to everybody that helped make this happen. I think we're really doing some high-impact work for a species that we really don't know a lot about. And it's already starting to make a difference. This lagoon has now been established as an important shark and ray area, and it's already contributing towards these really important decisions with Red Sea Global about coastal development and what areas are critical for this life stage. Thank you.
Thanks so much, Eloise. So we actually do have some time for questions, but since the next talk is also quite related to yours, maybe we'll go with Kaitlyn next and then tape some questions for both of you in the end. Yeah?
Okay.
So great, thank you. Next up with Kaitlyn.
Hello, everyone, and thank you for coming to my talk looking at the plasma metabolites in a critically endangered species, the Halavai guitarfish. And thank you, Ellie, for giving all the background on this species. So I wanted to start off by talking about how there's really not so much research done on this genus, the giant guitarfish. So as you can see in the top left, there's a pretty wide geographical spread of the rhino rays. However, most of this research has been done in Australia and the United States, and it's mostly focused on genetics. So this will be kind of the first opportunity between Ellie and I and other collaborators to look at in situ data. So because this species has not been studied extensively, a lot of the life history remains largely undescribed. And so using these plasma metabolites, we'll be looking into ontogenetic, sexual, and seasonal shifts within this population. And so we are looking only at the Alwaj population, which is located in the northern Red Sea. So as I mentioned before, we don't have much information on the rhino rays in general, and we also don't have much information on elasmobranchs in the northern Indian Ocean or the Red Sea. And so this extends over to metabolite information as well. Additionally, as Ellie said, this lagoon is undergoing rapid development in line with Vision 2030. And so these individuals are being caught at islands at different human construction phases. So to look at this, we're going to look, Ellie has already explained the movement part of this project, but we are going to be including nutrition, growth, reproduction, and heavy metals in the plasma, and genetics will be later on. So we did this by catching, measuring, and sampling blood from about 150 individuals. We centrifuged the blood, and this included sometimes using the car battery. We separated the plasma and froze this at -20 degrees, and then analyzed this data these samples colorimetrically using ELISA commercial kits on a spectrophotometer. And I just wanted to say a huge thank you to Raquel for showing us how to do this. She is working on tiger sharks in Brazil, and this kind of spurred our thinking on looking into this in the guitarfish as well. And had us apply for a small grant with the Save Our Seas Foundation. So thank you, huge thank you for supporting this work. So to go into this, we are going to look at 4 different biomarkers: nutrition, growth, reproduction, and heavy metals. So I'm going to start off with nutrition. And so I have to say with our nutrition, we've only looked at the young of the year and juveniles. Unfortunately, our kits arrived the day that I left. So we will be looking mostly at cholesterol, and that'll be the next slide. So triglycerides are generally energy storage, cholesterol, lipid transport, a steroid precursor typically for hormones, and longer-term metabolic state. And then ketones are an indicator of energy mobilization or fasting. Oops. And so as you can see here, as total length increases, cholesterol also increases. But when you separate that into male and females, you can see that there's a difference. So the females are, as they grow larger, there's more cholesterol, and the opposite is true for males. So this is possible that the females are starting to ramp up their systems to produce more hormones, but the males are focusing more on growing quicker. Next up, we're looking at growth. So we're looking at T4, T3, and the ratio between the two. So T4 is largely a precursor to T3, and T3 is more of the biologically active hormone. And so the relationship between these two is an indicator of metabolic and endocrine activity. And as you can see, total length increases, T4 decreases. As total length increases, T3 increases. So you can see as the individuals get larger, they are more actively converting this T4 into T3. Next, we look at reproductive hormones. So I was only able to look at estradiol so far, but all estradiol, progesterone, testosterone are all included in reproductive cycling, and each of them are important in different ways for the reproductive system. And so estradiol and progesterone roles are typically understudied in male elasmobranchs, and we are hoping to include this in our study. So I just wanted to give a little background on the Halibut guitarfish. Sorry for the slightly gory images. This was a fish individual, and she happened to be pregnant, and we were able to dissect her back at the lab and see what was going on inside. And so you can see that her right ovary is clearly dominant. And so this species exhibits aplacental viviparity, producing about 10 pups a litter from May through October in the Red Sea. And so we're hoping that our hormone work will be able to figure out what might be causing or the different stages of the reproductive cycle and seeing if there's any sort of seasonality in there. So with estradiol, we had the expected result of total length increasing and estradiol also increasing, but we can see also that males have a high amount of estradiol as well in their adult phase. Additionally, we looked into heavy metals in blood, muscle, and skin, and so we're looking at essential and non-essential heavy metals. So the essential heavy metals are typically used in biological processes, and the non-essential do not have a biological function, but are found in the tissues. And we are working with Chessie Mason over at Cardiff, and we're very grateful for her help. And in just looking at the initial results, I haven't had very much time to dig into this, but the total length increases with, or mercury increases with total length. And so we're super excited to look at how heavy metals are impacting the metabolites. And so this is super important because this is one of the first integrated studies on nutritional biomarkers with trace metal concentrations, as well as hormonal biomarkers, especially for this species and in this region. And so
The Rhinoprestiformes order is severely understudied, especially in terms of this, and so having at least a baseline for others to compare this to was very important for us. So future, we're hoping to finish our analyses, incorporate fatty acids, and then also include our movement and genetics data for a full profile of the individual sample. And I would like to thank everybody that has helped us. We've had a great team and lots of fun doing this research. So thank you.
Thanks, Kaitlyn. So we do have a bit of time for a couple of questions for either Kaitlyn or Eloise's talk that she just went before this. Does anybody have some questions? Yeah, we have one hand there.
Great talks, you guys. I was curious about, I have a question about the first talk about the Young of the Year residency. I was curious how you decided that they weren't predations and that they actually left and weren't resident.
Hi, thank you for your question. I mean, we don't know for sure, but we did accidentally drop a tag near one of the receivers. So we at least knew what it looked like when the tag kind of stayed within the lagoon and the animal was stationary. So that could potentially show what it looked like as a predation event. But yes, it's definitely possible. We know that the ospreys predate on them. We know that other larger fish can predate on halibut, but we don't typically see a lot of potential predators for them. It would also be possible that they accidentally catch themselves. We've seen that before, that they are stranded above the shoreline. But yeah, we were surprised by the results because ecologically we assumed that the smaller animals, more vulnerable, would be more resident than the juveniles. So yeah, and that's something that we're discussing in the nursery paper, and I'd be happy to share that with you as well. Brittany words are better than me.
Thank you. Maybe time for one more quick question, if anyone. Okay, we have one at the back.
Hi, thank you. Great talk. I was just wondering if you spread out your sample throughout the whole year to be able to observe this seasonality in the hormonal analysis?
Yes, so our individuals are actually the young of the year, and the juvenile blood samples are only from the fall and the winter, and then our adults are only from the spring. So we have a couple samples of adults from the fall, but it would be really great to kind of get that seasonality as well. But thank you for the question.
Okay, thank you. So we're going to now move on to the next talk by Garima Bora on guitarfish in India.
Good morning, everyone. My name is Garima, and today I'll be presenting the work that we've been doing to understand the ecology, fisheries interaction, and conservation challenges along the west coast of India. This work combines fisheries data, habitat surveys, local ecological knowledge to identify area-based management techniques that we could come up with to work with the fishing community. So we are working on these two guitarfish species commonly found in Malvan. First one is the sharp-nosed guitarfish and the white-nosed guitarfish. The sharp-nosed guitarfish is not protected legally in India, whereas the white-nosed guitarfish is a Schedule 1 species where you have penalties for being involved in trade and for catching these individuals. I would like to introduce our study site. It's of international and national importance in the sense that it's a very huge chunk of it is the Malvan ISRA, and if you look at it nationally, you have the Malvan Marine Sanctuary. This area is also of ecological importance because it contains a mosaic of habitats, and you have 3 major rivers falling into the sea, so you have a lot of nutrients coming in, hence a lot of productive fishing grounds. And where you have productive fishing grounds, you have heterogeneous fisheries functioning there, catching different kinds of fishes, and which of those are a lot of different types of elasmobranchs. Going into the methodology, to address the questions of where these guitarfishes are, we combined two complementary approaches. First, we used fishery-dependent cache data to understand patterns in the species, about where they are, which gear types are they caught in, different fishing locations, effort, and depth. Also, we looked at the fisheries independent way of monitoring these species, where we did beach transects to examine how habitat and environmental factors might have a role in the habitat selection. So this is the result of over 153 landing boats across a lot of different gears. And we found that both the species would occur in these gears and were caught throughout the year, but we had these specific peaks and seasonal peaks for white-nosed guitarfishes from January to April, and the strongest peak being around January, and whereas for sharp-nose, it was around December to February. We also looked at species-specific effect of these variables on their distribution. We found that gear type had a species-specific effect where you had sharp-nosed guitarfishes more being caught in trawlers, whereas you had wide-nosed guitarfishes being caught more in, by gillnet, I mean bottom-set gillnets and crab nets. So you have the wide-nosed guitarfishes being distributed across gillnets and shore scenes, which are a more near-shore fisheries practice. We also looked at site-level differences where we found the white-nosed guitarfish being more omnipresent throughout the northern sides of Malvan and the fishing grounds in the south, whereas the sharp-nosed guitarfish was more distributed northernly. We have no idea why, but we'll look into that later. We also compared the effort of the bycatch, which was like the results were kind of expected, where we had higher effort, had increased the likelihood of interactions with the gear. We also looked into how species were distributed across different depths. We found that the deeper you go, the more you run into sharp-nosed guitarfishes, and the more shallower you stay, you run into wide-nosed guitarfishes. Once we had this fisheries landing data, we wanted to explore what is actually happening in these near-shore areas. So we went to these beach stretches, and we started to look at what is happening at these beaches. And we did find these, we only found wide-nosed guitarfishes in these transects, and a random dog sitting in the water, which was one of the interesting species occupying these areas. Just to address why these species might be present in these areas, as we've only found juveniles and neonates, we haven't run into any adults in the 4 months of data collection. It's a pretty new study, but we have a video evidence of a wide-nosed guitarfish actively feeding. You see here, it kind of digs into the water, into the sand. So this is the result of the beach transects that we did. We found that certain sites like Tarashil, Tondavali, Saprebaganachra, which are a bit more deserted and you have very little human activity there, had more wide-nosed guitarfish detections per survey. We also had a very high correlation of site and disturbance level, so we clubbed 2 sites into these 3 different disturbance levels, low, medium, and high. We found a very significant difference in the number of people being present in these areas and the number of predicted guitarfish detections in these areas. We also looked into the socio-economic drivers and the local ecological knowledge that these fishermen have, and we had semi-structured interviews to collect this information. We were able to interview only 31 people as of now, fishermen, but we do plan to do more interviews in the next season. What we did find was size-based post-capture outcomes of these catches. So fishermen classify these guitarfishes into less than half a meter and more than a meter long. And usually the individuals which were more than a meter are either sold or consumed locally, whereas the ones which are juveniles and neonates are either discarded on the beach or released back into the water. But as you can see, the discard probability is really high. We also got to know that they do have quite a significant amount of contribution to the local economy. And given that the white-nosed guitarfish is protected legally in India, we had a lot of people saying that if you do want us to release it back into the water, you should provide reasonable compensation for that. We also looked into the local ecological knowledge that the fishermen had. We had a lot of people telling us about the species being associated around estuarine areas and sand flats. We also had a lot of people saying that normally in the high disturbance sites you do see them, but it's usually during the night when there's not a lot of tourists around on the beach and in the water. We also found evidence of depth-based size aggregations where fishermen talk about the smallest individuals being in shallow waters. We also found matching seasonal occurrence that the fishermen told us, which was matching with our ecological study that we had done with the fisheries dependent data. Also, fishermen talked about the declining trend of these species due to a lot of usual suspects like fragment degradation, overfishing, and bycatch. We also doing a lot of outreach and awareness programs in these areas because a lot of fishermen are not aware about the protected nature, and we don't want them to have run-ins with the forest department and get caught into the legality of all of that. Just to summarize what we've done for the past 3.5 years, I think, is we've found evidences of strong spatial and seasonal patterns in occurrence and catch. We also have evidences of gear being a key variable in dictating all of these bycatches. Depth and site influences also the distribution of these species. Juveniles and neonates are concentrated along the shallow waters more, and as of now we don't know what the adults are doing, and that is one of our objectives in the next season. The local ecological knowledge that we collected also confirms clear patterns in seasonality, habitat use, and size segregation, and economic dependence on these species. The next steps and plans is to understand the movement patterns using mark-recapture studies. We're planning to pit tag them in the next season, and exploring the change in habitat use due to local disturbances, due to tourism activity, and of people being in water and creating local pockets. And also long-term collaboration with the forest and the fisheries department, which are very huge stakeholders in the conservation of these species, and also involving the local people by developing a stewardship program, and also answering the question that I really want to know about is where are the adult white-nosed guitarfishes. Thank you, and I'd like to thank all the people and the local fishermen who've been really helpful for our study, and all the funders throughout the years, and my organization. Thank you.
Okay, thanks for the great talk, Garima. We don't have time for questions, unfortunately, but please feel free to speak to her during the break time and lunchtime. Next up is Yee-Hui THEN speaking about data poor rhino rays in Malaysian waters.
Hi, everybody. I know it doesn't sound like it, but I'm really excited to talk about rhino rays in Malaysia. For those of us who may not be as familiar, Malaysia comprises a peninsula as well as the northern part of the Borneo Island. We are bordered by 3 major water bodies, notably the narrow Straits of Malacca. That's one of the busiest shipping traffic lane in the world. We are, unfortunately, one of the top shark and ray fishing nation globally, and we take our seafood consumption very seriously as well. So when I started looking at working on sharks and rays in general, we find that we know very little about our sharks and rays, what species we have, what are the population trends, where can they be found, and most of the basic biology is mostly unknown. So over the last 10 years or so, supported by various small grants, we opportunistically sampled all across coastal Malaysia. And you can see that the sampling effort is mostly concentrated in the peninsula where I am based. So we updated the species checklist, questioned some questionable records, but basically one of the key things we found is that more than 50% of our shark and ray species are threatened. So we've published a number of work to close the data gaps for some of the more common species. And more recently, our lab started focusing more on rhino rays because of their ecological importance, as well as from what we're seeing in the field surveys. Sadly, like many others, sawfish also was last caught in 2014, so we think they're functionally extinct, and it is important for us to start doing something so that their distant relatives are not going to face the same fate. So supported by 2 funding sources, we asked some of these very basic questions. What species do we have, and what fishing gears are catching them? Where can we find the different species? How do different approaches in estimating relative abundance and fishing gear threat levels compare? And so based on the fishers' perception, what possible mitigation measures might they be willing to adopt? So in addition to surveying fisheries, landing sites, and market surveys, we also combed social media sightings and conducted interviews with both artisanal and commercial fishers. At the landing site, we also collect basic biological information and tissue for confirming of species identity. So while we cannot really get species-specific confirmation from social media sightings as well as interviews, we think that it is still valuable to make comparisons on how the relative composition might come out. So what did we find? From our fishery survey, the bottlenose wedgefish, Rhynchobatus australia, is the most common wedgefish species, and it's widely distributed across Malaysia. Most of these are caught using nearshore gill nets as well as trawlers, and most of the catch is actually of immature individuals. When we do find larger individuals, they tend to be females, and our estimated size at maturity is about 20 centimeters smaller than the available literature estimate, suggesting that fishing pressure is probably reducing their mean size. We, of course, also some morphologically ambiguous Rhynchobatus as well as Rhinobatos. And thanks to molecular techniques, we were able to confirm the presence of the eyebrow wedgefish, Rhynchobatus palpebratus, within Malaysian waters. Additionally, we also found the Ranong guitarfish, Rhinobatus ranongensis, in our waters as well. So this map shows you the different species shown in different colors, as well as the relative proportion of these different species. It's a bit busy, but I'm just going to walk us through quickly. So we find that Rhynchobatus australia is the most abundant compared to palpebratus, and for Rhynchobatus springeri, we've only found this once. And most of the time, we've never encountered the giant guitarfish, the Glaucus tigus. One of the notable findings is that the coastal waters of Kuching in Borneo is home to 7 out of the 8 rhino ray species. We found actually one dead female, Galápagos Islands, with 9 pups, suggesting that this area is important. And it's really nice that an independent study also shows similar results using camera images from trawlers. So please catch Liana's talk later on. Sorry, that will be tomorrow. And we also found that the waters of Johor is also important as it hosts 4 out of the 8 species. Now, when we look at social media sightings, it was interesting because we started seeing giant guitarfish. And like many of the previous presenters mentioned, they are quite often caught at sandy beaches by anglers. And we find that Galapagos typus is more abundant compared to Galapagos Islands. So this is a bit iffy, I know, but when we look at comparing the relative abundance between the 2 methods, they actually surprisingly agree very well for the wedge fishes. Now, on the other hand, the fisheries surveys suggest that there is a higher proportion of the smaller guitarfish, the rhinobatos, but the social media sightings show the importance of giant guitarfish. So based on the proportion of animals caught in the different gears from a fisheries survey, it certainly showed the importance of the trawlers in orange catching all of the rhino ray species. When we look at social media sightings, it's showing the importance of hook and line, particularly recreational anglers, catching, in fact, most of the rhino ray groups. So the fisher interviews corroborate the importance of the trawlers, but also a high proportion of the gillnet fishers are also catching the different groups of rhino rays. So from our interviews, hook and line is associated with the lowest animal mortality. And most of the fishers agree that rhino ray catch has been decreasing, and the main reason is overfishing. So when we surveyed a number of mitigation measures, most of the, well, not most, but a bit more of the fishers agree that they prefer live release of females and the juveniles. So the key takeaways is that with the 2 new species record, we can confirm, with the exception of the sawfish, we have 8 rhino ray species, and the hotspot is in coastal Kuching, Sarawak. And they have distinct fine-scale distribution. So different methods was important here to reveal the importance of the different gears. And live release is possibly the mitigation strategy that we should look at next. So there's so much things left to do, but one of the things that we hope to do is to involve people as part of the solution, maybe even rewilding as well. So Amanda talked earlier about rhino rays, looking at it with BRAFS and eDNA. And please listen to Irshad's talk later, talking about the eyebrow wedge fish. And with that, thank you very much.
Thanks for the great talk, Amy. Again, we don't have much time for questions, but please do catch her at lunch.
Hello, hola, sí, venga. All right, and this works like this. Yes. So here I introduce you to the main character of the talk today. This is the common guitarfish, one of the 2 different species of guitarfish that can be found in the Mediterranean Sea. And of course, like many species of this session that we are talking about, their declining population status has promoted the protection of the species by almost all different protection figures that can be found in the region particularly. Unfortunately, these regulations might have just arrived way too late for the western side of the population, and in particularly 30 years late. That has been the amount of time that the species was thought to be extinct in the Western Mediterranean. And this is something that we thought for a long time, until in 2023, everything kind of changed. And it's because in this little corner of southeast Spain, sightings like the ones that you can see here allowed the rediscovery by science and the conservation, because of course divers and fishers knew that they were there. What they did not know was that it was critically endangered and thought extinct, which allowed the declaration of the area as a reproductive ground and as an ISRA. No? So after celebrating that we rediscovered this species in the region, the next obvious question was, why here? No? And the first hypothesis that came up straight to our minds was from the very famous MPA that can be found in the region. So this is known to be one of the most successful MPAs, all well enforced, and specifically this one is managed by the Regional Fisheries Administration, which has really made the fish biomass in this region to boom. So if these guys are really expending most of their time in this area, could explain why they have disappeared elsewhere, but still they are here. Knowing this, it's not only out of curiosity or scientific minds, it's also very important for the conservation of the species, because despite the rest of the area, it's a Natura 2000 and an environmental MPA, it has absolutely no fisheries regulation. This means this is the wild, wild west in terms of fisheries risk for these protected species. That's the reason that in 2024 we initiated an acoustic tagging campaign. I don't know if it will be possible to remove the sound because I forgot to do that, even though it was in the guidelines. So basically we did an acoustically tagging campaign in 2024, 2025, where we captured the animals through diving because the visibility is really good and the animals can be found between 10 and 20 meters. We didn't want to, it's a protected species, to make the work easier in terms of ethics. It was easier to do it this way than by fishing. And it's a very similar technique that what they used in the Angel Shark project, but the thing is that we actually bring the animal on board to place the tag internally. And this is so because the dorsal fins of the animals are very thin, so we were not confident of putting the acoustic tag on that dorsal fin. As well, this allows us to take as well blood samples, and we also take genetic samples of the organisms, put a visual tag that in, well, 2 days ago we got a resighting of some of the animals that were captured in 2024, seeing that they've grown 10 centimeters and they still retain the visual tag. But we've already seen that some of these animals lose the tag because they still have the little scar of the acoustic procedurement. So we also do pit tagging. So far, during these 2 past years, we've been able to capture 60 different individuals. From those, 34 we've tagged because they had enough size to do that safely. And we deployed a whole network of 20 receivers to have a little bit of a trade-off of coverage of the MPA, but as well try to detect any kind of movement within the ISRA and outside the ISRA, as we have sightings all around this area that you can see here. So a little bit here, what I will be introducing to you is the data from 2024. We did not know anything back then. We thought that we may have found like 2, 3, 4 guitarfish. We thought that they were only in the area of Cabo de Palos. So we will need to incorporate the data of 2025 because thanks to fishers' support, we discovered other hotspot areas that we could go to reliably tag these species. So with this in mind, know that those individuals tagged in Cabo de Palos, incredibly resident, barely use other areas of the ISRA, really just transition zones. So great, that means that potentially they are using a lot the MPA. Well, these animals are only using 0.3% of the MPA according to our array because the majority of the time they are spending their time in the shallow areas, which are not covered by this fisheries MPA. Luckily, in the upcoming 2 weeks, there will be an expansion in that yellow area that you can see here that will increase to 22% of the detections inside this fisheries MPA. But still, up to 70% of the detections that we have this 2024 will be located in an area with no fisheries legislation, but with the potential to have so, as we are talking with a Natura 2000 MPA that requires management plan. So with all of this, we cannot ensure that individuals do not move between one area to another, as we've been able to already see organisms that were tagging one and another area. And well, this has promoted us to want to let the different environmental and fisheries administrations know this preliminary data, because decisions are being taken at the moment in relation with the expansion. It can matter for these organisms if it has fisheries policies associated, and as well as right now the environmental MPA is trying to find ways to improve, right, this instrument. As well, it's very important to know what we are not detecting, what we are not seeing, and in particular, we are seeing that the bigger organisms are not detected as much in the area. In particularly, this one disappears in the winter months to be again in the summer, right? So we started a little bit of a pilot study with the satellite tag, and because again, the dorsal fins are just way too thin, we did a little bit of research doing CT scan and necropsy, and ended up deciding a modified method from Yang et al. From 2025. Instead of using the metal wire, we use non-absorbable sutures to kind of like have the tag well attached. Still, we will not be able to know the results until December next year, because it's been programmed for 1 year, and we need more information in case this doesn't work, especially because we need to figure out why are they still here. And we think that the answer may be in deeper waters, and it's because of this reason that thanks to Fundación Biodiversidad, we will be working 1.5 extra years to try to figure out the habitat use of these species in deeper habitats using BROP and ROP. And luckily, if we manage to find the logistics to get ourselves some acoustically released receivers, we will be able to detect also the animals that will be emitting until April 2027. So with all of this, we don't stop. We want to know as well, we need information to be able to define what we need to do for the survival of the species in relation of distribution, where they are and where they are not, as well as abundance. No? And for that reason, we are still collecting community science sightings that are defining not only important sites for common guitarfish, but other critically endangered species in the area. All of this we are validating using as well diving censuses and doing a very strong awareness raising campaign in terms of for the diving community, but as well for the fishers, and a lot of Instagram. If you are interested, please follow us. We don't want to be influencers, but we know that in order to tackle the biggest threat for this species, that at the moment is that people do not know it is threatened, that it is thought to be extinct in the area, we need to generate that knowledge. We need to generate the belonging, and I love it because we did not organize these kind of sculptures that you are seeing here. It's been adopted by the community. So with this long term, it's important to incorporate all this information into a conservation action plan, so it doesn't depend on a PhD student like me and the team in the University of Murcia to develop the guidelines and the next steps for the conservation of these species. So if you want to know more, follow us on Instagram, keep in contact, because during the next 2 years, I will need to be submitting thesis. We will be posting publishing a lot of the results, if everything goes well. Now I need to sit down and actually analyze and write to kind of, keep getting information about these species and promote some conservation change for the long term. So thank you very much.
Thank you very much, Maria. Please feel free to catch her later on the breaks if you have any questions. Next up, we have David Ebert with his talk on guitarfish.
Hi, everybody. So to get started, I have a kind of a pop quiz question for everybody. Does anyone under 35 know who the guitarist is? Anybody? Not named Rachel Aitchison? How about anybody over 35? Okay, well, catch you afterwards. Talk about Eddie Van Halen later. So, the talk today involves a bit of a journey that started 40 years ago for me when I first arrived in South Africa to work with Leonard Campagno. Amongst the first projects he gave me, assigned me about 6 weeks after I arrived there, was to go out and to look for a particular guitarfish. So I found myself standing in a field near Bushman's River, near Kenton-on-Sea in the Eastern Cape, waiting for a South African Defense Force helicopter to come pick me up and take me out to Bird Island. Bird Island happens to be this area where this one particular guitarfish was caught and described in 1926. And since then, that species has never been seen. This is the holotype of that species. It was originally described as Rhinobatus ocellatus. It's now Arteriobatus ocellatus. And this is the holotype at the British Museum. Now, guitarfish taxonomy is kind of complicated. It's fairly convoluted in a lot of areas. So one of the things we had to do was to see if we could find this thing. And over my time in South Africa and the years since, I've gone through thousands of guitarfishes trying to find this particular species. One of the problems is, if you go to the literature, the identification is relatively vague and often misleading. This particular publication by John Wallace in 1967, which is a monograph on the Batoid fishes of southern Africa, which is a classic reference. If any young people are not aware of it, you should be, whether you live in Africa or not. It's a classic reference. However, in this reference, particular thing regarding the Arteriobatus ocellatus, which was described as Rhinobatus, he had this picture here and said at the time it was the first specimen seen since 1926. Well, as it turns out, this is not ocellatus. This is actually a variation of Rhinobatus annulatus, which is known commonly as the lesser sand shark in South African waters. And the species now, as I mentioned, is Arteriobatus. Well, the problem is when sometimes, and this is a very common thing, it gets kind of caught up in the literature, is misidentification. And since that, this thing's passed its way on down through the years, always being the same thing, being misidentified. And here you have, in Last et al.'s Rays of the World, you have, with a wonderful illustration by Lindsay Marshall, you have over there the lesser guitarfish or sand shark, Arteriobatus annulatus. And then here you have what was illustrated as a speckled guitarfish, which I don't think is a really good common name for it, but Arteriobatus ocellatus. Well, the problem is that's not ocellatus. And I talked to Lindsay, who's a friend of mine. She provided me with the reference material, which was some specimens at the South African Museum, which coincidentally I'd looked at many years ago. And the illustration is based on another color variant of Arteriobatus annulatus. So for better part of 100 years since this thing was caught, there was no real confirmed identification of this thing. And the reason, not just this particular species that I'm using kind of as a proxy for guitarfishes in the Western Indian Ocean, but for just guitarfishes, but also for guitarfishes in general, is that guitarfishes in almost anywhere you go seem to be a staple in many countries of the world. And over the last few years, I've been to like South America, I've been to Asia, Indonesia, Africa, and every place I go, guitarfishes seem to be a staple there. But many cases, like this particular Malagasy guitarfish here, this young Malagasy fisherman caught, was an undescribed species that I actually described a few years ago, Arteriobatus andisabini. And the thing is, we've got these species that we're trying to like develop policies for protecting, and we don't even know what they are, let alone looking at like their life history, doing any kind of ecological or fishery studies on them, especially when we don't even know what they are called. So myself and a team of researchers from South Africa, Mozambique, Tanzania, as well as from Canada and the U.S. have been looking at this question of these different guitarfishes. And we're working with both the Rhinobatus and the Arteriobatus, the two genera. But I'm just using, again, the Arteriobatus as an example, where you have this particular genus, which at one time they were all in the one genus, Rhinobatus, but they've been split. But you'll notice they have a very limited geographic region, most of them. And some of the species, like the Malagasy guitarfish down around Madagascar, it's just endemic to that area there. You'll see the distribution of several of these other ones being very limited. So given this high rate of endemism within these things, it's even more important to be able to identify these things. So we could tell what species we have in these different regions. So fortunately, in the last few years, we've been able to get specimens through collaborations with research groups in different countries, work with WCS, Rhett Bennett's group, as well as the Nansen surveys they've done through FAO and the Norwegian stuff up and down the east coast of Africa. And from this stuff, I should see if this is a pointer here, but you notice as we ran all these things here that Rachel here, who insisted I put her picture up here, because she's a little bit of a narcissist, but she ran these really elegant statistical analysis, and down here, we had these 4 fell out here, but these 2 turned out to be Acteriobatus omanensis, and these other ones here turned out to be kind of an unusual species. Now, one was the holotype that I just showed you. Another was a specimen that came in on one of the Nansen surveys. We had a whole bunch of them identified as Acteriobatus ocellatus, but we really weren't sure, were they really ocellatus or not. When we looked at them, most of them were not, except for this one exception here, that there's a sense down there that we looked at. And so that prompted us to do some further investigation, looking at both the morphological as well as the genetic, and looking at some statistical analysis on this particular species. And what you can find in this image that came out a little blurry is that we found one specimen here in this whole group that nested in with the omanensis, which turned out to be distinctly different, and which turned out to be, we believe, is actually a Acteriobatus ocellatus, which was very informative to be able to know that we've actually found these particular species. And what ho, after nearly 100 years of not seeing this species, recently the image analysis unit at the South African Institute for Aquatic Biodiversity was doing some filming, ROV footage, off East London in the Eastern Cape, which is about 150 kilometers from Bird Island. And they captured some usual, like the typical lesser sand sharks, but they captured this one particular image here, which clearly, if you think back the holotype I showed you, has these patterns of these sort of gray-blue spots across the back here. And this, to my knowledge, is the first confirmed record of this species in about 100 years. And so this thing actually, in a kind of a way, it's kind of really set me off my own personal journey to look for lost sharks. And after 40 years of looking around for this thing, it's finally nice to kind of actually get some confirmation of this thing. But now, this was an in situ specimen that we came across and we were able to identify and confirm, comparing to the holotype. But what was even better is we were able to, off the Nansen surveys, we got one specimen of all the ones that were labeled as ocellatus. One specimen came in, and that was the genetic one I showed you that also fell out statistically different. And this is actually a true Acteriobatus ocellatus. And this one came off of southern Mozambique. So to date, we've got 3 specimens, Bird Island, East London, and southern Mozambique for this thing. It's a very cryptic species, and we're not really sure much about it. It's very distinct when you see this thing, but it's really important in terms of like being able to, now that we've been able to identify, 3 specimens, but to learn a little more something about them. And so as we try to like investigate, and this kind of is really, we're just starting out here, because with these taxonomist stuff, we're actually learning, we've discovered like 3 new species in the last few years that we're in the process of naming. There are a number of other new species that we've also been able to identify. But the real reason why it's important to really be able to identify these things so we can develop better plans is, if you look at here with the sawfish are in the same order as the guitarfishes, the Rhino pristiophores. And the thing with the guitarfish is that, in recent years they've been implemented some conservation policies to try to protect this species. But this species actually largely disappeared from most of the Western Indian Ocean, and it's almost at a point where it's too late that this species will ever actually fully recover. Similarly, the other guitarfishes may also be disappearing and fading away with very little notice. And so really to be able to like look at the, look at the guitarfish before it's almost too late with the policy is going to be very important over in the next few years. And so you really have a situation where you have these guitarfish that are, like say, they're disappearing, while we play for time, and then these things, is this really going to be like the last dance for these guitarfish? And thank you very much for your time. This is our team here. And thanks, Save Our Seas.
Thank you so much, Dave. Amazing talk. Unfortunately, we don't have time, but I'm sure people will come and get you.
Good afternoon, everyone. One sec. Firstly, I would like to say what a privilege it is to share this space with so many inspiring researchers and storytellers from all over the world. It's really a special place to be here. My name is Candace, and I am presenting on behalf of my colleague, Niall O'Reilly, who unfortunately couldn't be here today. Our project was looking at the population dynamics of a critically endangered white-spotted wedgefish in South Africa. So the rhino rays are a very unique group of elasmobranchs. They are characterized by their shark-like ray appearance, and they have these flattened heads and ventral gill slits of the rays, and yet they also possess these very muscular and powerful tails and these prominent dorsal fins like that of sharks. So there are 68 known species of rhino rays, and 80% are already categorized as either vulnerable, endangered, or critically endangered, and 48% are facing high risk of extinction. So in the last 30 years, we've seen almost an 80% decline in most of the populations of rhino rays, and this is largely because of the high value of their fins, and these are sold obviously in the shark fin trade. They also are very slow growing and late to mature, with some species taking anywhere between 5 and 12 years to reach sexual maturity. They live in these coastal and inshore areas, which also puts them at risk of high fishing pressure. So our project focused on Rhynchobatus judensis. This is a white-spotted wedgefish whose range goes from the southern Africa all the way up into the Persian Gulf. And I wanted to just highlight some of the local threats that we have within South Africa. So this species faces the risk of transboundary overfishing. So while it might be protected within marine protected areas of South Africa, as soon as it moves northward into Mozambique, it faces high fishing pressure, where a lot of it goes unregulated by artisanal and commercial fisheries. We also, unfortunately, in South Africa still have shark nets. So between 1980 and 2017, we had more than 2,800 individuals that were caught in these bather protection programs. Since 2005, South Africa has started to transition to using drum lines in hope of reducing the amount of bycatch that's been caught. But of course, this is a whole nother topic to talk about. In South Africa, we also have a very high recreational fisheries. So this species is very targeted by sport fishermen. It's a very competitive industry. And of course, if proper handling techniques and depending on fight times, these animals are at risk to post-release mortality, to physical injuries, and capture-related stresses. Lastly, we have habitat degradation and loss. Because of the way these animals use the coastal and inshore areas, they are at risk of things like pollution or coastal development, and also siltation from the runoffs from the estuaries. So these threats, combined with their biological sensitivity, it makes them really vulnerable to these pressures. So our study site was in the Isimangaliso Wetland Park. This is a UNESCO World Heritage Site, and it's actually part of South Africa's largest marine protected area. It spans 220 kilometers down the coast, all the way until the border of Mozambique. And it has been a designated ISRA, which is amazing. This is because you can find, it's an important area for reef manta rays, for feeding. We also have a very healthy population of the critically endangered spotted ragged-tooth shark. And we also have a lot of other megafauna that uses these areas. So this park is a really important migratory corridor for animals that move from South Africa towards Mozambique. The aims of this project were to describe the demographic profile of the white-spotted wedgefish within the Isimangaliso Wetland Park, and to investigate the influence of environmental and seasonal variables on their presence. So for our sampling, we actually had another big ongoing project where we were conducting a baseline assessment of the elasmobranch community in this park. And from this, we extracted the white-spotted wedgefish data. So we used stereo bruvs, and we actually built these bruvs ourselves, which was quite a fun experience. And believe it or not, Pythagoras and trigonometry actually do come in handy after high school. So our sampling design was kind of a blanket sampling across the whole park, and we targeted different depths from 8 to 35 meters. Upon deployment, we also took a number of environmental variables, including temperature, current speed, current direction, swell height, and depth. And then later into the modeling, we included lunar illumination and seasonality. This is a video of a very beautiful adult female wedgefish that we saw on one of our BRUV deployments. And the BRUVs also managed to capture a lot of year of the young and neonate wedgefish. So for our image analysis, we use an open source computer software called i3S. And this program you can use with any animal that has a unique spot pattern. So for example, whale sharks or ragged-tooth sharks. And we can create a database of all the different individuals using this software. And we can then identify if there's been any resightings or what happens over time. This allowed us also to look at the max number of individuals rather than just using max N, which is better for rare species. Because the BRUVs are stereo, it means that they are specially calibrated using a software called CGIS, and from this we can actually measure the individuals and classify them into different life stages. So over the 4 years, we deployed almost 800 BRUVs, and we had about 162 white-spotted wedgefish observations. What was very interesting is that we saw a female biased sex ratio of 1 to 4.5. And this was very interesting because it actually challenges the data that is collected through the recreational fisheries, which is mostly male-dominated. So there could be potentially some sort of spatial segregation based on their sex, and it would be interesting to explore this further. We also successfully measured 42 individuals. And what was nice about this is that we found quite a big size range, ranging from year of the young, since white-spotted wedgefish are born at around 60 centimeters, and we had some really large adults, just over 3 meters. So this kind of highlights this area could be used by these species which are known to be highly resident, and they could actually maybe complete their entire life cycle in this park. When doing some GAMs, we modeled the GAMs to look at the presence. We found that looking at lunar illumination, that the presence and abundance of wedgefish decreased as the lunar cycle increased. That we found no sex-specific seasonal shifts within the park. And when modeling the Julian day, so this is when we take the months of the year and we transform it to look at cyclic seasonality, we found that it was a strong predictor. And we also saw that there was a clear increase in activity during midwinter and early summer. Finally, temperature had a very low predictive power, although presence did tend to increase with rising bottom temperature. So just to summarize our findings, it was very interesting because we actually found the highest number of white-spotted wedgefish ever recorded on BRUVs, which validates this method to study the species. We also established a very clear demographic profile. There was indication of seasonal and environmental responses. And finally, we could identify this region as very important to study the species. So in conclusion, the Isimangaliso Wetland Park can serve as a critical habitat. It offers a very unique context to study these species because of the reduced fishing pressure. And it's amazing that we have this data poor species that can be found in such high numbers within the park. So moving forward, I think transitioning from a video-based approach to incorporating acoustic telemetry and incorporating data from the recreational fisheries could be really interesting to further understand like the full movement of these animals around the southern African coastline. So thank you very much, and
thank you, Candice.
Yeah, thank you.
Fantastic talk. We have about a minute for a very quick question. So if we have a question from the audience. Okay, thank you. I'm sure people will find you in the break. Now, I would like to request Ardukan Askin, and Ardukan would be talking about photo identification database for guitarfish and wedgefish.
Hello, everyone. I see a lot of people standing in the back. Don't be shy, come grab a seat. It's really exciting to be here on behalf of my entire team from MIARU, which is the Maldives Shark Research and Conservation Program. So today I want to take you guys through our work that we started just this year to identify rhino rays and all of the other key habitats that they inhabit all across the Maldives. Candice has given a great introduction already, and she also happens to be wearing the matching top to my pants. So I'm really excited to have a South African twin. But yeah, as Candice mentioned, guitarfish and rhino rays in general are among the most threatened group of elasmobranchs, and they've had an 80% population reduction over the last generation. So that means that within our lifetime, they're at risk of going extinct. Our grandkids might not be able to see them, and it's really their life history that makes them so incredibly vulnerable, and the fact that they also inhabit very shallow areas which tend to overlap with coastal activities and fisheries. In the Indian Ocean, there's a lot of targeted fishing activity for rhino rays. Mostly for their fins, which have a high value in the shark finning market, but also for their meat and skin, which is used and consumed by coastal communities. And for bowmouth guitarfish, for the jewelry and amulet trade, their horns are used. Who knew about this? Who's hearing this for the first time? Put your hand up. For me, this was also a surprise. Here you can see some of examples of bowmouth guitarfish jewelry made from the horns captured by one of my fellow National Geographic explorers. Now, within the Indian Ocean, there is one place that stands out as different to all, and that is the Maldives. It's very exciting for me to be here and hear everyone's work using sampling methods, collecting skin samples, biopsies, stomach content analysis, because those are all methods that we haven't yet been able to do in the Maldives. And the reason for that is because the Maldives is a national shark sanctuary and one of the only countries in the entire region that is not fishing guitarfish and wedgefish at all in any shape or form. But it's also the geography of the Maldives that makes it incredibly unique and a key habitat and an important refuge for these species. 99% of the Maldives is water, and the majority of the country consists of 26 atolls with shallow lagoons on the inside, so perfect sandy habitats, and fringing reefs on the outside of the atolls. Here you can see the bathymetry, and you can see all of these atolls from above with the satellite imagery. So it's an incredible place for these animals. And it offers a variety of different habitats: shallow reefs, deeper fringing reefs, sandy, muddy areas, mangrove forests, and seagrass. So we have a wide variety of all the things guitarfish and wedgefish need to thrive. We have two species, the bowmouth guitarfish and the bottlenose wedgefish. And if there is the person here that named them, I have a message from the Maldives because we would like to request a different name. The locals call them Muddy Miaru, which I think is the best name possible. It means shark ray in the local language. But despite the fact that we have these species, and they've been regularly and continuously cited by divers and in large aggregations, we have absolutely no data. We know they're there, we don't know where they are, what they're doing in different areas of the Maldives where they reproduce. So this year, we launched the official Spotting Rhino Rays project in the Maldives, which is the first ever concerted effort to study these species. Now, I mentioned that there's no targeted fishing, but that doesn't mean that they're in the clear. We do have some really severe threats: habitat loss, which I'll explain a little bit more, climate change, and pollution. The Maldives is a country that is currently rapidly expanding its land reclamation, driven by this idea that because land is so scarce, there has to be more that we make. So Ras Male is a new project that's just recently launched. It's a giant mega land reclamation project. And when I say land reclamation, what I mean is that massive dredgers pump up sand from the seafloor to create artificial islands. And the majority of this happens in already existing lagoons and on top of coral reefs. Here you can see an island that's filled up the majority of its lagoon to come to existence. And the dredging leaves literal scars in the country's geography when you look at it from a satellite image. So not just do we create these artificial islands, and all of these islands you see here in the image are artificial, but it also leaves these massive dredged channels. And this means that we're directly and very rapidly losing key habitat for the guitarfish and wedgefish, because reefs and seagrass meadows are buried under sand, and the sedimentation and smothering pretty much impacts everything. So we've started to do a multi-method approach combining dive surveys, aerial surveys, archival research, so looking into historical sightings, and citizen science to gather data on guitarfish and wedgefish. Why citizen science? It's our biggest tool. Millions of people come to dive in the Maldives every single year, and thousands of locals are employed in the diving and tourism hospitality industry, which means we have so many eyes and ears on the water. And given that the country is 800 kilometers long, that's the same distance as the south of France to the Netherlands, it's impossible for us to survey every place. But if we have people going anyway, we can mobilize them. So we launched the Maldives Shark Hub, which is our national citizen science platform, where users can send sightings, mostly dive guides, fishers, and tourists. When we receive a shark sighting or ID, it comes into our system. We can then verify it and cross-check it against our ID catalog. And for this, we use, like Candice explained, the spot pattern. We also use the dark markings and also the color. We've noticed that some individuals have different colors. Here again, just a quick visualization of the primary areas that we focus on for ID. Same for the bowmouth guitarfish, and the markings come in really handy. And then we also run this through AI and several softwares and algorithms to get the IDs we have. And in most cases, even manual identification. And our citizen science program has yielded some incredible observations. This one is a video taken by one of our team members, Sarah. And what we're hoping to do with the IDs is to really track and monitor individuals over time and start to get this baseline information. For every user who sends in a shark that's new, we also send an ID card. In this case, this was one by our team members that we named after Dr. Sylvia Earle. And it's an easy dashboard that tracks everyone's activities. It's a fully integrated data system. So for every bowmouth, guitarfish, and every wedgefish, any data ever submitted by our team and citizen scientists is available at one click. And this includes all the images and videos. So far, we have compiled over 100 historical records, more than 138 guitarfish sightings. We've conducted 241 dive and drone surveys between the team and identified over 50 bowmouth guitarfish and over 95 bottlenose wedgefish. Most of them we see at tilas, which are these formations that you see in the middle. If any of you want to know more about the amazing reef formations of the Maldives, come talk to our team. And we've also been receiving observations of mating interactions, which is incredibly exciting because it helps us to pin down reproductive areas. Now, what's next? We want to really dive deeper into the reproductive work. We want to identify where these animals go to reproduce, where the year of young and neonates spend their time, how they move between the atolls, and how much they're actually at risk of the land reclamation happening. And we launched this project at the start of this year, combining all of our dive surveys and everything from the previous years. And something that was really interesting is that last year there was a really cool paper published about incidental bycatch of bowmouth guitarfish in pelagic fisheries, debunking the myth that these animals only spend time in shallow waters and don't really travel larger distances. So our question is, are they really safe in the Maldives Shark Sanctuary, or are they leaving our waters, which would put them at further risk? So if any of you here are working on these species and want to come talk to us and our amazing team, please come find us, and a huge thank you to everyone.
Thank you. Thank you very much. Fantastic talk. Unfortunately, we don't have time for a question, but finally, a sawfish talk. We have now Alberto Mejia Paniagua, and he'll be talking about the cultural significance of sawfish in Honduras.
Thank you. Hello, everyone. You can hear me? Thank you for being in this fantastic session. I want to tell you, English is my third language, so I will do my best. Also, I want to share this has been my Roman Empire for the last 10 years. I'm truly obsessed with the sawfish and their history. And also, I'm a scientific illustrator, and all the illustration and drawing you will see made here is made by me. So if you want illustration, please let me know. Oh, sorry. So why this is important? Because probably you know, sawfish are the most endangered groups of constrictors in the world. Also in Honduras, we have the two species, Prisis pristis and Prisis pectinata. Prisis pristis is considered extinct in the Pacific by the USN, and in the Atlantic, we still don't know how much it's about the present and their distributions. So, well, across the sawfish distribution, many coastal cultures have the sawfish as an important part of their worldview. So, for example, in Australia and some part of Africa, Panama, also Brazil. For Brazil, I would like to invite you to the presentation of my friend Mika Sherbet in this room as 420. He gonna tell more about the project in Brazil. So maybe you think my map is kind of wrong, but I really want to show the importance of the Global South as a region for science and knowledge production. And maybe we should start seeing our ideas like more upside down. So I have this question for Honduras. Is there any traditional significance of sawfish in Honduras? Are there any sawfish left in the country? But where I did to start. So in Honduras, we find La Mosquitia, one of the most remote areas of the country. It's considered one of the 5 most important areas for biodiversity and conservation in Central America. La Mosquitia is a large wetland with a vast system of interconnected lagoons, brackish lagoons, and a long mangrove system along its coast. So if I were a sawfish, I want to live there. And because in the past 5 years, we received reports of sightings, encounters, and incidental captures by fishermen in the region. So we start by asking questions. In Spanish, we say asking you can go to Rome. And this makes sense because this is my Roman Empire. So we want to understand the economic importance and the most recent sightings and the importance of the sawfish has in the mosquito culture. So we made 143 response surveys in 7 communities along the Mosquitia coast. So our first question, when was the last time you saw a sawfish? Most people never seen a sawfish in their life. Even me, I never seen a sawfish in my life. But we have an important number. At least 8% saw one in the last less than a year. So in this group, we asked if they was target fishing or by catch. So also we asked them if the animal was used for. Many they say they don't they has not consumption, and another part, my water, say only the rostrum was used. Is this because many rosters were painted and used as decoration in their homes and churches? And also, to understand the main dangers of the sawfish in the region, we asked what type of fishing gear they use. And also, honestly, if you look like a brush in the water, they may be tramalet as your worst enemy. So it's easy to see why tramalets were the main fishing tool using to catch them. So for answer the question, sorry. When was the last time a sawfish was seen in Honduras? The most recent sighting of sawfish was in May of 2025, sorry. But this year I've been in La Mosquitia and I see a pregnant woman moving a big boat. So I think maybe she needs help. So I go and she asks me, what are you doing here? So I say, I work with Lily and we work with sharks and rays and me with sawfish. She say me, oh, we catch a sawfish in October. So we have we have record for the last year, and that makes me very, very happy. So during what details we identified during our research? We identify a possible causes of population decline in the region by incidental capture, commercial trade, climate change, and the legal gaps. We identify a possible trade route for the rostrum from a Mosquitia, Honduras' main sawfish source, and the loss of cultural ritual importance. So we identify that we are lost in a species, at the same time we are lost in history and ancestry. So what we to do next? We want to start eDNA monitoring to improve our conservation efforts. So if any donors or people who want to collaborate, please let me know. And start a citizen science program so we can have real-time records and continue with the cultural revival. So this is some of what we do. We are painting a mural with a large image of the sawfish to keep the image of the sawfish in the public imagination. We also make a festival here. Held a festival, we are highlighting of the sawfish, and we open a channel where the fishers can report and their sightings and records. So for finish, I want to tell about the importance of the sawfish into the other world for the mosquito people. The mosquito people believe we have three deaths. The death of the body, the death of our soul, and the third death and the final death. The image of the sawfish, also, sawfish is called twaina in mosquito. So this image appears for the great mother sawfish, and in this moment, the sawfish stay with them and the third death. So because the mosquito people believe they become to rain. So thank you. I want to say Tinki Pali, which means thank you very much in mosquito. Thank you for our donors, and thank you especially for the mosquito people for giving us the permission to tell her history with you guys. Thank you.
Thank you. Thank you so much, Alberto. We have about 40 seconds for a quick question. So if anybody has a question, we could very quickly take it. Yeah.
Please make louder.
Here.
And
Hi, mates. Nice presentation. By the end of today, let's talk about the eDNA stuff, right? Okay? I can help you a bit of the eDNA approach that you want to apply there.
Yeah.
Thank you.
Fantastic. Thank you very much. Thank you, everyone. Thank you, Alberto. Fantastic storytelling.
All right. Okay, I'm guessing you've heard of shifting baselines several times already this week. It's the idea that we measure normal based on what we've seen in our lifetime. And the marine world looks very different. Sorry. Okay, we're starting again. Okay. Okay, the marine world looks very different to us than it would to this family of fishers right here. And that's because, wow. I'm going to move on. Okay, an issue commonly faced, particularly with non-targeted species, is that we're very data limited. And we're asking this question late in the game. However, a number of potential sources exist that can fill in these gaps, particularly when pooled together. Today, we're going to apply this strategy to sawfishes, a group of shark-like rays that, due to the shape of their iconic rostrum, are easily entangled in modern gear types. They live in shallow coastal areas likely to interact with humans, as you can see by the subadult green sawfish here, and have been historically collected both as opportunistic bycatch and targeted for the rostral-headed fins. Being long-lived family of relatively low fecundity, their populations are slower to regenerate, and they are now extirpated from approximately 60% of their former range. Due to habitat fragmentation, current blind spots in the distribution make it difficult to assess or monitor them in vast northern Australia, and as bycatch historical record keeping is very scarce or absent. So let's go back to that question again. Where can we find historical data? How about the trophies themselves? And this works because the species can be easily ID'd from the saw itself by the size and the spacing of the teeth, and from the rostrum alone, we can determine the total length of the individual and used to estimate the maturity, or long-term changes due to overexploitation. And this is really handy as the body of the animal is obscured in many of the historical photographs we come across in both the SARA database and museum collections. So Sharks and Rays Australia has been collecting sightings within Australia for the last 10 years, which I built on by visiting over 50 museums and historical societies during my PhD across Queensland and New South Wales. Many of which were smaller than this room, which is feeling really big right now. And when visiting these museums, a lot of them, especially the local ones in particular, will include information on the family, gear, the date, and even a newspaper article. And the National Library of Australia has digitized thousands of articles from regional and metropolitan papers from 1800 to 1954, where it's capped due to copyright. A quick search for sawfish returns about 4,000 articles today. Integrating these different data sets allows us to piece together a more complete timeline. After filtering for duplicates, of which there were many, we were left to 350 records spanning 1875 and 1990. And I'm happy to chat with anybody further about this, but it was based on many existing matrices in the archival space. And binning these occurrences into 15-year intervals, we can see what starts out as a capture hotspot, basically where the people are, down by Sydney, and then it slowly moves up. And if I bring in the species composition alongside, you can see right away that the vast majority were green sawfishes, which is in green. And they were especially common between the border between New South Wales and Queensland. I have for reference on the right in that big black box, their current distributions, which is based on my second chapter, that shows the extent of range contraction here in that time for all species, except for narrow sawfishes in blue. A really important note here is that Noxopristis and Pristis clavata, which are in the blue and yellow, are very poorly represented in the historical literature, which makes sense if you know them because they are the smallest of the sawfish species, and therefore were not nearly as newsworthy. And if I focus here, just on this inner box of the highest density of our reports, we produce noticeable decadal trends. Early warning signs across abundance, which are shown as the bars, and the maximum total length for that bin, which is the points overlaid on top. The three northern regions and the upper facet still hold sawfishes today. So the trends in the total length depression there are not significant, and they're actually more indicative of a changing species composition, which I have as little boxes at the top of each of those graphs. The three bottom, however, do show significant declines over time, which follows that trend of we have an increased catch, then that drop during the 40s, World War II, and then an increase again, and about that same time, you see the total length decline. And in those immediate years, a few things are happening. These are the gear types that were used per decade. And what I'd like to point out is that shift towards trawling. As part of the industrialization of fishing from the 1940s, where we see vessels increase in size and efficiency and go further offshore and becoming the norm up the coast of New South Wales and Queensland. So where does that leave us? Extinction timelines highlight this a bit. I use 4 different models, and I include the 2 top regions. There are still sawfishes, although they are not nearly as common and they're less frequently cited today. And I included them so that way we could have a test of that this methodology wasn't incorrectly predicting sawfishes were extinct in an area where they still are, and it passed. But in contrast, those ones further down in Brisbane, Gold Coast, Northern Rivers, they were all extirpated by, by the 1985. Something else to notice though, the Mary, which is further north, had been eradicated far earlier. So to investigate this further, you actually exploit this location in particular. And so bringing all this together, we're seeing size selective harvesting as a key player here. Taking the biggest individuals really makes sense from an economic standpoint because you get more bang for your buck, but that also means you're removing the largest, most successful breeders, which decreases the reproductive potential of those populations. And as preserved parts fetch even higher prices per kilo, and be sold for longer, species like sawfishes that are in that big red box are in that perfect storm. You add in catchability and lack of regulation, and the process is essentially accelerated as we've seen here. So in summary, Priscidae was a depressing and excellent case for fisheries-induced effects over time, where 2 of these largest species were removed from over half of their range in about 100 years. This archival approach lets you see it playing out in a record that's directly transferable to monitoring other species by looking at range, size, and reporting frequency, building the pieces of the puzzle. Of course, there are still limitations. Namely, we saw the best results for the largest sawfish species, which has the biggest percent, the saw is the greatest percent of its body and therefore the biggest trophy. And it was definitely aided by the digitization of media. This would need also a preserved structure. However, chondrichthyans have many of those as a possibility. So while sawfish are an extreme case, the pattern is not. There's a lot of things I didn't get to get into. If you want to hear more about human attitudes towards sawfish and text analysis, or why the term saw shark is the bane of my existence, I'm happy to chat more. And I could have really spent the whole 8 minutes on this slide due to the number of people that have been instrumental in this moment right now, my supervisors, all the scholar volunteers who cataloged and downloaded everything, the citizen scientists who reached out with their family photos, the museum curators who either welcomed me into their collections or literally digitized things and sent it through, the Save Our Seas Foundation for funding my travel, and honestly all the senior researchers who took a back seat at this conference, who allowed ECRs and students like me to have a stage today. So thank you.
Thank you, Nikki. Great talk and perfectly on time. So we have time for a question. Quickly, if we have a question, we can take it. Nope. Great. Great talk. Thank you. Now, we'd move on to our next speaker, Miko Shervett. And Miko would be talking about challenges of sawfish conservation in an Amazonian coast.
Hi. I hope everyone's excited to hear a little more about sawfish. I'll be talking about the challenges to sawfish conservation in the Amazonian coast. So 2 of the 5 species of sawfish are present in this region, Pristis pristis and Pristis pectinata. They're all critically endangered and protected under CITES Appendix 1. These are coastal, marine to freshwater species, and we have historical and current records of them in this region. Here's a little overview of the area covered by our project, spanning from Guyana through Suriname, French Guiana, and parts of northeastern Brazil, where we see ideal habitats for sawfish. This is a highly productive region with large stretches of mangrove forests and some unique habitats associated with the Amazon River discharge. However, it's extremely understudied. which is something we need to address as this is one of the last potential strongholds for sawfish. So a little bit about the importance of sawfish to this region. Sawfish rostral teeth are a preferred material for cockfighting spurs, which is still a cultural practice in parts of South America. And the sawfish rostra are also used as decoration and trophies, and sometimes thought to bring good luck. Also sometimes used in traditional medicine. And similar to what we heard from Alberto a little bit, sawfish are important to the indigenous people in the area. For instance, we've spoken to people from the Caripuna tribe, where they call sawfish barata or aheche, and you can see an art piece made by one of these people with a sawfish. So getting into the challenges here, transportation is a big one. A lot of these places are pretty remote. We have some slightly bigger cities, but when we're traveling along the coast, we see a lot of roads like this, a lot of boat travel. That picture of the road is from a 17-hour bus ride that we took. And that means we need a lot of funding. It's a lot of traveling, difficult places to get to. And there are also not a lot of local researchers here, and the people that are here end up being spread out pretty thin, and they can't really focus a lot of resources into something as specific as sawfish. This means that almost all of our information is coming from fishers and local communities, which, as we know, requires a lot of trust. And that's also difficult because we don't have anyone permanently stationed here. But we have still been able to get some pretty good information speaking with the fishers and the locals, including how many different common names there are for sawfish here, with there being many countries that speak many different languages in a relatively small area. We have all these different names just here, and it's easy to get confused when we're trying to do interviews or trying to share information, with some of the names referring to different animals in different places. Regulation is also a big challenge. There are limited resources and so much water. It's difficult to regulate all of the fishing, and that's even harder with the political instability, especially recently in that region. A lot of borders are being disputed, and that makes regulation harder. Also, only Pristis pectinata is protected in the national legislation in Guyana and Suriname, when we know both species are here. We also see a lot of illegal or under-regulated fishing, and there are a lot of large fishing vessels coming in from other parts of the world to fish in these waters, which ends up driving local fishers into the neighboring countries. And so that's a big issue here. The size of the teeth also makes the trade harder to regulate. They're pretty easy to hide, and move around without being caught when you're dealing with just the teeth and not the entire rostrum. The sawfish teeth can also be confused or passed off as different animal parts, like a jaguar tooth or a caiman tooth. They all look pretty similar, and without training, the enforcement agents might have a hard time telling them apart. So what do we know? Sawfish are still here. They are still sometimes getting captured, and these incidental captures are frequently fatal for larger individuals that are more difficult and more dangerous to release. And we know research and conservation are very difficult. This means we need to really work with the local governments to build better informed and more effective legislation. And we also are working on training enforcement agents to better recognize sawfish parts. And we also have to work with the local communities, trying to teach people a little more about sawfish, especially the younger people that haven't really seen sawfish in their life, and it can grow a sense of ownership and responsibility and pride in their local resources so that they can take over conservation and connect knowledge to protection. And really, we need more research. There is still hope for sawfish here. We still see them there. We just need to keep fighting. So I'd like to thank our funders, the Shark Conservation Fund and Save Our Seas Foundation, and also Sharks International for having me here and letting me present. Yeah, thank you.
Thank you so much, Mika. Such great work. So thank you. We do have time for a quick 2 questions, maybe. So if anybody has. Okay, if not, I'll ask a question. Fantastic work and in such challenging conditions, and then to present it in such nonchalant way. I mean, so cool. Absolutely inspired. So what made you still continue to do this work, although the conditions were so difficult to work with? What was your inspiration, and what would you do next?
Honestly, I really love working there. It's a lot of fun, and it's very rewarding to kind of go to places where people don't really go and work where people haven't really worked. Yeah, we have been doing some eDNA work and a lot of interviews, and we want to keep going back there. It's just been a little bit more difficult recently with all the political things happening. It's been a little dangerous, but we hope to get back in there soon, do some training, do some more field work. Yeah.
Thank you, Mika. Okay, that is the end of our regular talks. So we're now going to move into the speed talks, starting off with Valerie, who will be talking about remaining hotspots for rhino rays derived from grubs.
This guy? Okay. Hi, I will be presenting a set of data from the Global Finprint Project. I just wrapped up coordinating Global Finprint 2. I'm a little new to the rhino ray world because I do live in the US, but we had all this data and I figured we're on the right side of the world. So Global Finprint, as many of you may know, is the world's largest shark and ray survey on coral reefs. This data set is from the early 2010s up to 2025 at this point. I'm going to be highlighting the critically endangered the critically endangered and large-bodied Rhinidae families and Glaucosticidae only. And the rhino ray presence is noted as max N, which many of you are familiar with. Maximum number of species in one field of view at one time of the same species. So this would be max N of 1. If there was 2 of the same species, would be max N of 2. But for the whole data set, we only have max N of 1 because, again, these guys are critically endangered. So we have now 30,000 hours of video over 58 jurisdictions worldwide. And the species composition that we have seen thus far is very few bowmouth guitarfish and giant guitarfish, less than 10 combined. More of the bottlenose and white-spotted wedgefish in the 40 counts each, and 151 total from these two families. Most are not ID'd to the species level because of their very similar morphological characteristics. Please note the active orientation of the animal to the brev. They almost always circle it. So in our survey, there are 36 range states. These guys are missing from 23 of those, so they're only, for our survey, utilizing 36% of their range, which is not very much. There's low abundance of these in 10 countries, and they're only common in 3. Our hotspots include South Africa, Mozambique, Australia, and they're quite spread across Australia. So we have Kimberley Reef, Pilbara, Ningaloo, and the Southern Great Barrier Reef. And all of these are marine protected areas or partial marine reserves. The percentages of animals that we saw per brev are as up on the screen. Again, they're critically endangered, so we're not seeing very many anywhere. And then an interesting case study, apart from Global Fin Print Project, is from this paper by Conrad Speed at Ashmore Reef in Australia. And in 2004, they saw 4 individuals from 46 brevs. And then this was a marine protected area in 2004, but it was not protected as such. It was kind of a paper park. In 2008, the Australian government started defending it with a vessel, and boom, we had an increase, 14 individuals in 89 brevs deployed. So Ashmore Reef may be the hottest of the spots. This is another case in Ponta de Oro in Mozambique. I really just want you to focus on the first 2 columns. In the 2010s is in blue, and the 2020s in orange. We had significant increase, and over the same time period, the Isimangaliso in South Africa had the same trend occur over the same time. So, brevs appear to be a good sampling tool, and in areas with good visibility, obviously. An anecdotal note that we observed was that they prefer areas of low relief, vertical relief, so sandy bottom or low rubble, because they are a ray. And in conclusion, we have presented evidence that some MPAs are working in the right direction for rhino rays, and brevs are a useful tool for surveying them. If you know of any other hotspots, please feel free to email me. Thank you so much to all of the collaborators. Global FinPrint is such a huge, overreaching project, and to the funder, Allen Family Philanthropies. Thank you very much.
Thank you, Valerie. Our next speaker will be Nala Khan.
Okay, nice. Hello, I am Nahla from Bangladesh, and today I'll be talking about how we attempted to trace the species-specific biology into somewhat of a management pathway in Bangladesh. So our work is titled as this, where we determine the age of wide-nosed guitarfish from their vertebrae. We build the growth models, plot their maturity. We already know that most of the rhino rays are super threatened with extinction, and their conservation and management suffer the most because of data scarcity, which is quite prevalent in regions like Bangladesh. This particular species, the wide-nosed guitarfish from the rhino ray group, is assessed as critically endangered globally by IUCN. However, this species is frequently captured and brought to the landing centers along the coast of Bangladesh, and no life history studies of this particular species is present as of today. Thus, we worked on this with the objective of developing a low-cost age determination method and building their growth trajectories to understand their life history and provide the information for species-specific conservation. Our methodology was quite simple. We bought the already captured specimen from the landing center, then we took the morphometric information and we took the vertebrae out of it. After processing the vertebrae, we simply used wet sandpaper to polish the vertebrae and get the thin vertebral section, which is extremely cheap, rather than using the quite expensive diamond-bladed saw. The diagrammatic workflow is presented here, and we calculated or interpreted the annual growth bands as age and fitted them into growth models. And the maturity was examined by examining their gonads. So we found out that the wide-nosed guitarfish at the Bay of Bengal grows up to 110.5 centimeter. The females attain larger sizes than males, and the maximum age counted was 16 years. Then we fitted the observed length and age data into the growth models, and our study suggested that among the three popular growth models, the von Bertalanffy, Gompertz, and Logistic, the von Bertalanffy growth model describes the growth of wide-nosed guitarfish the best. So the von Bertalanffy growth parameters here also fall into the posterior summaries in Bayesian approach at 95% credible intervals, which also reinforced our model support for von Bertalanffy. And for maturity, we found that the wide-nosed guitarfish matures around the age of 6 years. The length at maturity was 62.2 for males and 69.8 for females. So which is quite low, so they're slowly maturing. Coupled with their low growth coefficient found from the growth models, we can confirm that this species is a case-selected species like other rhino rays. Studies like this can actually tell us about how species biology are making them more vulnerable to the stressors they're already exposed to. But overcoming challenges that we faced, like we couldn't include any neonates, we could not conduct the age verification or validation, overcoming those challenges, more robust studies like this can actually inform species-specific conservation and management, we believe. I am thankful to the Save Our Seas Foundation for funding my work, and I'm also thankful to the Mortimer Laboratory and again Save Our Seas Foundation for having me here and funding my travel. I am extremely grateful to the team at Bengal Erasmus Lab. I couldn't have done this alone, so a huge shout out to my team. Thank you, everyone. Let's make the ocean lively again.
Hello, everyone. I'm Yara Grossmark. I'm going to present a short part of my PhD work where I wanted to test actually what if marine conservation started with encounters and not only with where the animals are. So conventional conservation deals with where are the animals, where do they live, where do they stay, where do they feed, where do they breed. But my offer is to look at who meets them as well. Okay, what do they do? What is the type of the encounter, how and where? So this is
a place where conservation could be a little more fine-tuned. You can start looking at if there's a conflict, or maybe this is an opportunity to create coexistence. And I'm offering that human dimensions should actually be data points instead of an outreach after the plan. So what did we do? We took a case study of 2 critically endangered guitarfish that are very common in Israel, the black-chin guitarfish and the common guitarfish. And we used mako citizen science observations from 2014 till 2022. Those observations consist of a photo, a date, a location, but more importantly for this work, they also consist what the people were doing while they encountered the guitarfish. The reports were then validated by experts to identify the species and life stages. And we also added an activity layer, what the people were doing. And then we entered it into a spatial model to see if it stands in a test in the targets that we set. Oh, sorry. I'm missing one. Okay, I'm missing the results for some reason, sorry. So the results, so I'll just talk about them. So we also did a gap analysis, and the first thing we noticed was that the young ones were actually, the juveniles, were the one that were least conserved in the Israeli protected areas in the Mediterranean. And we also saw that different life stages aggregated in different areas and actually met different activities of humans. So the young ones were really very prominent in anglering and in beach walking, while the older ones were only seen while diving or snorkeling. So that means that actually when you look at it, so maybe the conservation should also include these activities. You should maybe do gear restrictions to protect the young ones, and maybe just restrictions on the amount of people diving in a certain area where the adults are aggregating. So what I'm offering is that conservation areas should not only be about protecting habitat, they should also choreograph coexistence and offer a way forward for humans and guitarfish, or actually any marine wildlife, to coexist together. I'm currently looking for a home for a postdoc to continue this work, so if you're interested in human-wildlife interactions and conservation planning, come talk to me, and thank you very much.
Thank you, Yara. We do have time for one very quick question, if someone has one. No, it looks like you're off the hook. Thank you. Next speaker will be Kayla McCulloch, who will be talking about environmental DNA for a critically endangered wedgefish.
Hi, everyone. So, yes, I'm going to be talking about species-specific environmental DNA for critically endangered wedgefish. So as we know, trying to detect rare and elusive marine species can be extremely challenging, especially when we know almost nothing about them. So species-specific environmental DNA coupled with droplet digital PCR, or ddPCR, is one of the most sensitive tools that we have. This is because droplet digital PCR partitions each reaction into about 20,000 droplets, and this allows us to detect down to a single copy of DNA in an environmental sample. And so this is a reliable and quantitative tool that is more robust to inhibition than other methods of eDNA, including qPCR and metabarcoding. And so because of this high specificity, we wanted to use this method to try and detect the extremely rare species, the clown wedgefish. So the clown wedgefish, Rhynchobatus cooki, wasn't described until 2016, and at that point, only 11 specimens had been seen. And at that point, it had been considered to be possibly extinct for almost 2 decades until a single specimen was rediscovered in a Singapore fish market in 2019. And so prior to this study, we had very little information about the distribution and habitat use of this species, with the only clues coming from social media reports that indicated that this species was found potentially in the waters surrounding Singkep and Lingga Islands in Indonesia. So our approach for the overall study to try and detect the clown wedgefish used a multi-pronged approach, and the first part was environmental DNA. And so in June of 2023, we collected 3 replicates of 1 liter benthic water samples across 33 sites surrounding the entire circumference of Singkep Islands in Indonesia. And these were collected at shallow depths from about 0.5 meter to 8 meters. And we covered all habitat types, including coral reefs, seagrass beds, mangroves, and sandy bottoms. And then I developed a species-specific CO1 ddPCR assay to be able to detect the clown wedgefish, and this was fully validated against all species of Rhynchobatus and other rhinopriciforms. Unfortunately, after screening all DNA samples, we had no detections of the clown wedgefish, but because of the high sensitivity of this method, it is highly possible that this species just was not utilizing the coastal habitats where we sampled. And as I mentioned, the overall project used a 2-pronged approach. So the second part that we did was fisher interviews that were conducted by our team member, Benaya Simeon, who presented on that earlier this week. And so our results kind of showed that no detections from eDNA in shallow coastal habitats, but through the fisher interviews, they are still being caught, so they're not extinct, yay. However, they are being caught at depths greater than 20 meters, so outside of our eDNA sampling range. And so future efforts should kind of focus on areas in deeper habitats, specifically in the area between Singkep and Lingga Islands, which are known areas where the species is caught, and then also a little bit further north off the coast of Sumatra, where we have additional social media reports of these wedgefish being landed. But overall, the study shows that eDNA plus ddPCR is a highly sensitive tool that can be used for rare species detections, even when there are actual no detections. So I'd like to thank our entire team, including those in Indonesia, and our funders. Thank you.
Thank you, Kayla. We have time for a question, if anyone has a question. Yep, Maddie Green up the back there.
Run all that way. Hi.
Hi.
Thank you. That was really interesting. I really like you going with the digital droplet method. How were your limit of detections for your in-lab testing?
Yes, our LOD was 0.05 copies per microliter, so very low detection rate. So if it had been there, we should have detected it.
Okay, thank you, Kayla. Now we're going to stay on the theme of eDNA with Christina Nardini.
Hello, everyone. I'm Christina, and today I'm going to be talking to you about our Sharks and Rays Australia eDNA project with sawfish in Queensland and Cape York Peninsula. So sawfishes are among the most threatened marine fishes globally, having undergone severe population declines and range contraction due to bycatch, low reproductive rates, and habitat pressure. Today, Northern Australia represents one of the last remaining global strongholds for 4 of the 5 species of sawfish, yet our understanding of their current distribution remains incomplete due to their rarity and the difficulty of serving in remote and turbid habitats. In this study, we used environmental DNA to assess sawfish distribution across Queensland with a focus on remote regions. Our goal was to better resolve current distribution patterns and evaluate the effectiveness of eDNA as a broad-scale monitoring tool in these systems. So we collected eDNA samples from 86 sites across freshwater, estuarine, and coastal habitats between 2018 and 2025. So that's over 400 replicates that we got. Sites were selected based on historical records, known habitat suitability, and accessibility for us. We applied species-specific qPCR assays targeting mitochondrial markers for these 4 sawfish species that you can see labeled up here. All detections were validated through sequencing and strict contamination controls, and methodology for this study was created by Maddie Cooper at James Cook University. So detections were strongly concentrated in northern regions, particularly within the Mitchell River system, Gulf of Carpentaria, and Princess Charlotte Bay. P. priestis, where you can see them, the red dots on the map, were the most commonly detected species, with detection split across freshwater and coastal systems, including multiple positives in the Mitchell River. A. cuspidata, the blue dots that you can see on the map up here, showed the broadest distribution along coastal sites extending down eastern Queensland. P. zizeron, the green dots on the map, were largely restricted to Princess Charlotte Bay with a single detection in Port Douglas South. And P. clavata was not detected in our samples. Overall, 50 sites returned no detections, as you can see by the black dots on our map, with the highest proportion of negatives occurring in central and southern Queensland sampling regions. So these preliminary results align with established biogeographical patterns, showing that sawfish become less abundant towards southern Queensland and are increasingly confined to relatively undisturbed northern coastal systems. The groupings of detection in Princess Charlotte Bay and the Gulf of Carpentaria highlight their importance as key pupping habitats for these species. The absence of P. clavata wasn't surprising, as few of the sites that we sampled were in their known habitat. And for P. priestis, detections aligned with its known life history, including juvenile use of freshwater systems such as the Mitchell River, where we have confirmed sightings and catches. We didn't see P. zizeron in central Queensland, which was a little bit surprising, as we know their distribution is known to extend farther south than that, so that's something I want to look into when I'm doing my further studies on it. And these results highlight the value of integrating eDNA into broader monitoring frameworks. However, considering eDNA's limitations, it should be considered as complementary to other sampling methods. So I would like to acknowledge the support of my collaborators, funding partners, and field teams, some of which are in the audience today, if you want to say hi. And as well as our Aboriginal land and sea rangers and local communities who contributed their knowledge and access through this work. We really could not have done it without them. And I want to give a massive thank you to Barbara, who has been the best support. I would not be up here without you, so thank you very much. Feel free to reach out after or anytime. So.
Thank you, Christina. We might need to save questions till after. So. Okay, our next speaker will be Alistair Harry. He'll be talking about sawfish in Western Australia.
Hi everyone. Today I'm going to talk about trends in catch rates of sawfish on the Australian Northwest Shelf, and I'd like to begin by acknowledging my co-authors from Fisheries Western Australia who were involved in this work. So this study was published in 2024 in Endangered Species Research, and it was a reconstruction of catch and analysis of catch rates from logbook bycatch data for a trawl fishery that operates on Australia's Northwest Shelf, which has historically caught between 25 and 80 sawfish a year as bycatch. As you can see, the fishery operates in this red box here. It's quite far from shore, and these are adult sawfish that are caught by the fishery, green sawfish and narrow sawfish. I was really excited to learn that my organisation was collecting this data, because typically we don't have species-specific data for sawfish, and to my knowledge, this was the first species-specific indices of relative abundance for these species anywhere globally. And the other unique thing about this data set was that we were also able to verify about 20% of the records through onboard deck cameras and fishery observers, so we had a pretty good idea that they were accurate. And catch was, the catch rate itself were analysed using fairly standard generalised linear mixed effects models to standardise catch per unit effort. So what did we find? I guess one of the really key findings from this study was that since 2006, the catch rates of the green sawfish, which is the much larger species and the more abundant, have doubled. Now, this isn't a population model, so we don't know exactly how this translates to abundance, but it certainly is a positive finding. And I think in terms of interpreting this, it's important to look a bit back, look sort of back in time to understand what we might be seeing here. Between the 1960s and the 1980s, the particular region circled in red where this fishery operates on the Northwest Shelf was a major fishing ground globally. At one point, there was projections that up to 1 million tonnes a year of demersal fish could be caught from this area. At the time, or between the 60s and 80s, it was fished intensively using indiscriminate fishing gears by foreign fishing fleets. In 1979, Australia declared its foreign fishing zone, Australian fishing zone, and the foreign fishing fleets left, but the Australian domestic fleet moved in. And we know that the Australian domestic fleet also had a large bycatch of sawfish. But I guess what's quite unique about this particular situation is that since 2010, almost all of the fisheries that have bycatch with sawfish in this area have stopped operating, and even this particular fishery, which had just 3 vessels left in it, has now been permanently closed as of the end of last year. So what we could possibly be seeing here is a recovery of green sawfish following a possible historical decline. And I think I guess the key finding or sort of key message is that while we might not be able to confirm that exactly, we can say that even in this area, which has been one of the most intensively trawled parts of the Western Australian coastline, we still have these 2 species present, which is obviously extremely encouraging. And I'll leave it there. Thank you.
Thank you, Al. Now, we do have time for a quick question. If anyone has a question for Al. Okay, thank you, Al. And our last speaker for the session will be John Carlson, who's also talking on the theme of recovery.
Thank you, Mick, and thanks everybody for sticking around to the last talk of this session. And I want to congratulate the session organizers, because I thought this was really informative for the lesser than studied elasmobranchs, but as Irene points out, flat sharks are important too. Just something really quick that I want to make sure everybody awares of, that everything I say from this point forward is my scientific opinion only and not anybody else's. So that's all I'm going to say. So I think everybody knows this. Small tooth sawfish, their status is not very good. But I do want to highlight in this slide that there are still some potential viable populations in the western North Atlantic, primarily in the Bahamas, Mexico, Cuba, and the area I'm going to talk about in the United States. So shifting gears for a second and just talking about marine protected areas, we had a symposium earlier on protected areas and on ISRAs and how valuable they can be. They have been shown in some cases to be very valuable in protecting not just elasmobranchs but other species. And I think Nick Dolby first brought this concept up of MPA serving as a lifeboat, where populations of sawfish could be reseeded from these lifeboat areas. So that's kind of what I want to focus a little bit on, is the potential for some of these areas to reseed areas where sawfish are gone or extirpated. So the area I want to talk about is an area that's pretty much been my home for the last, since about 2009 when I started this work, following the work that Colin and Tanya Wiley did. In this area is the southwest portion of the United States, in particular Florida. For those that are not familiar, Miami is over on the, I don't want to hit the wrong button and shut everything off, but Miami is over on the right side of the map. The Southwest Florida area contains two critical areas of sawfish habitat, one being the 10,000 Islands Everglades National Park, and then one to the north is the Charlotte Harbor. Now, in the 10,000 Islands Everglades National Park, this area represents a very robust ecosystem. The mangroves are very dense. The prop root system is very inclusive of very tight, thick mangrove habitat. And actually, you can see down here, this was in the picture in the middle, you can see with the circle, there's a sawfish swimming along the edge, and you can see how dense this habitat is. So within this habitat, since the late 1980s and 1990s, there have been several surveys, 2 scientific surveys conducted, and 1 survey conducted by the park, which interviews anglers, collecting information on catch, effort, location, things along those lines. So what I did was, with the hypothesis that if we saw any type of increase in the population, we'd see it here first, rather than anywhere else. So what I did in a little scientific methodology is I took all the data that was available and modeled it in a generalized linear model. First modeling what's the probability that the animal was caught. You know, there are certain factors, location, time, gear, things like that. And then once that first probability was met, the second probability was to model the rate at which they were captured. And those 2 abundance indices independently are brought together into one index. But we're not done here yet. So then what I did from the data sources that I had was apply those data sources into a Bayesian model, which takes all of them and accounts for both process era and sampling era. And this is what we get in the trend. As you can see, beginning in the late 1980s, the trend is relatively flat, but then around 2009, it starts to increase. So we're starting to see an increase in abundance for this population or subpopulation of sawfish in the Everglades National Park in 10,000 Islands. So how does this compare with that other area of critical habitat that I pointed out to you? Well, in this area, it's a lot different. This area has been subjected to habitat modification. Most of it is coastal development with a lot of condos. People, when they Google Florida and they get pictures of condos, you get pictures of this type of area. There is still some natural habitat left, I should point that out, but overall it's been severely modified by both freshwater inflow and the overall structure of the ecosystem. So then when we look at the 2 abundance trends, this was done independently in a study by Farmer, you can see the blue, the index for the developed area is going down, but the index for the protected area is going up. It's big contrast. So what does this mean is that we do have evidence that these protected areas do provide, in those areas that again are largely protected from habitat modification. And I should also point out too that commercial fishing has been banned in these areas since the 1980s, although recreational fishing is still allowed. But I believe that dense, relatively pristine habitat is what these sawfish are using to help recruitment and expand their population. Now, one thing to point out is that we are beginning to see in other areas, and again, this is Florida. On the left side is Tampa Bay Estuary, and on the right-hand side is the Indian River Lagoon. And through our public encounter data, we're beginning to see sawfish reoccur in those areas, which suggests that we're starting to see that reseeding from the lifeboat in Everglades National Park in 10,000 Islands. But, and this is a big but, those 2 areas have a lot of anthropogenic modification as well, and in some areas the habitat has been severely degraded. So what this tells us is that if sawfish are going to start recovering in these areas outside that quote-unquote lifeboat, we're going to need some significant habitat restoration in those areas. So I'll end on that, and I kind of wanted to show a little, there he goes. All right, this is a video that was taken by Michael Scholl when he came out with us. What's up? So you can kind of see a sawfish working its way along the edge of those habitats just to see how they influence, and they'll work their way in and out of those mangrove prop roots. So I don't know if I have any time for questions, Vic, but if you want to email me or catch me after during the break, thank you very much.
Thank you, John. That was fantastic. Has anyone got a quick question for John before we finish up? No, I don't think so.